How did scientists discover that the Earth had undergone ice ages? Evidence of ice ages comes in three different forms: geological, chemical, and paleontological. Each of these provides scientists with material to develop a general understanding of the effect of ice ages over the past billions of years. Geological evidence corresponds to the stripping of rocks, the deposition of unconsolidated material, sculpted valleys. Geological evidence has its limits. They can be deformed or erased over time, and it can be difficult to date them accurately. Chemical evidence is defined by variations in the isotope ratios of fossils found in sediment and rock samples. The isotope ratio is the ratio of the number of atoms of two isotopes in the same sample. To study the most recent glacial and interglacial periods, scientists use samples of ice cores and ocean sediments. They may also contain air bubbles, enabling analysis of the composition of the atmosphere during the period under study. Finally, paleontological evidence is characterized by changes in the geographical distribution of fossils. Most organisms that live in warm conditions do not survive ice ages, whereas those adapted to colder temperatures are more likely to survive. Thus, a low fossil count in higher latitudes indicates the spread of ice caps. Scientists rely on these three types of evidence and on numerous data sources to obtain the most consistent results possible. One of the most valuable sites used as an indicator of ancient climates is the Antarctic ice cap, which has provided information on past atmospheric temperature and composition. Little Dome Sea is a drilling site set at an altitude of over 3,200 meters, or 10,500 feet, above sea level on an East Antarctic plateau. Temperatures regularly drop to minus 50 degrees Celsius or minus 58 degrees Fahrenheit. The landscape is carpeted in white, and the first coastline lies more than 1,000 kilometers or 620 miles away. Since 2021, scientists have been drilling into the ice cap to extract ice cores 4 meters or 13 feet long. The aim is to reach ice layers at a depth of 2,700 meters or 8,900 feet. The idea is to be able to go back in time 1.5 million years and reveal new information about the climate of the time. 90% of the world's ice is concentrated in Antarctica. Antarctica covers an area of 14 million square kilometers. This gigantic white continent is an invaluable archive of the Earth's history. Its ice, thousands of meters thick, contains information on its temperature and the composition of its atmosphere. Scientists are as interested in the ice itself as in the air bubbles trapped inside it. The latter reveal the concentration of various greenhouse gases, such as carbon dioxide, methane, and nitrous oxide. Drilling has enabled us to go back a long way in time. What is the origin of these climate changes? Various causes are responsible for the appearance of ice ages. Some of them are interdependent. The Earth's orbit around the Sun undergoes cyclical variations over time, known as Milankovitch cycles. During these periods, the Earth's distance from the Sun and its inclination will change, resulting in a redistribution of the sunlight received. The movement of tectonic plates influences the glaciations that the Earth undergoes. Depending on the position of the continents, the flow of warm water towards the poles can be disrupted or blocked, leading to the formation of ice caps, variations in the amount of continental and oceanic crust affect winds and ocean currents. Atmospheric compositions such as carbon dioxide and methane concentrations is linked to ice cap movements. Greenhouse gas levels decrease with their advance and increase with their retreat. Let's take a look at the 5 major ice ages in Earth's history. $2.4 billion years ago, the Earth experienced its first ice age, which was also one of the longest in its history. This was the Heronian glaciation. This glaciation began 2.4 billion years ago and lasted until minus 2.1 billion years during the Paleo-Proterozoic era. The oceans occupy almost 25% more than their present volume due to the degassing of the Earth's mantle. Lava from volcanic sites is rich in potassium and phosphorus, making it a high-quality fertilizer. Due to wind and weather, lava naturally breaks up and crumbles. Small pieces of material embed themselves in the fractures of the magmatic rocks and accumulate there in large numbers. As the rocks are porous, they retain not only moisture, but also a warm temperature ideal for the development of life. In the end, these seemingly inhospitable places were the scene of the development of the first organisms. Among these microbes carried by wind, weather, or tides are cyanobacteria, a type of prokaryotic bacteria with no nucleus. These unicellular organisms can live both in water and on land, in large colonies. Their metabolism is based on photosynthesis. They produce oxygen. Cyanobacteria in the air transform nitrogen into ammonium and nitrate, while those in the oceans trap carbon and produce the limestone of stromatolites. The combination of lava fragments and cyanobacterial growth gradually forms soil. Then, little by little in these same privileged environments, more complex organisms such as mosses and fungi appear. The latter will develop a method of rooting, in particular, mycenes. It was thanks in part to them that life first walked the Earth, since by breaking rocks they enabled the first terrestrial settlements. It was only later that pollen also contributed to the proliferation of life. The Earth is not yet very conducive to the development of more complex forms of life. It is covered with active volcanoes, and toxic clouds are permanently present. Vast expanses of lava cover the ground as the sun floods in its deadly ultraviolet rays. Although cyanobacteria remain in the water, the flora is still widely dispersed. Nevertheless, a major event occurred 2.4 billion years ago that changed the face of life. It was the Great Oxidation. At that time, oxygen in the Earth's atmosphere was scarce. It remained trapped in minerals or enclosed in water. The Great Oxidation affects the Earth's waters, biosphere, and atmosphere. It can be seen as a catastrophe or an opportunity for life on Earth. Most organisms are anaerobic, but cyanobacteria are beginning to develop oxygenated photosynthesis. They occupy a new ecological niche and reproduce very rapidly by harnessing the sun's energy. Photosynthesis produces a waste product, oxygen, which cyanobacteria expel into the air. Initially, this oxygen is absorbed by the oxidation of surface iron, but as the number of cyanobacteria increases, the oxygen sinks, becomes saturated. This accumulation of oxygen in the atmosphere has had devastating consequences for anaerobic organisms, which cannot survive in the presence of oxygen. Oxygen is a highly reactive element that can disrupt the metabolic processes of anaerobic organisms by forming free radicals. These unstable molecules can damage proteins, nucleic acids, and cell membranes, leading to cell death. As a result, many of these organisms have been eliminated, leading to a mass extinction of anaerobic organisms. But increased oxygen levels have paved the way for the evolution of new life forms better adapted to these conditions. Oxygen enables much more efficient cellular respiration than other forms of energy metabolism. In other words, organisms capable of aerobic respiration can produce far more energy from a given quantity of nutrients than their anaerobic counterparts. Some organisms have evolved to use oxygen as the final electron acceptor in their electron transport chains, thereby greatly increasing the efficiency of ATP production, which could be described as the cell's energy currency. In addition, antioxidant enzymes such as superoxide, dismutase, or catalyses have evolved to neutralize toxic free radicals produced by reactions with oxygen, enabling these organisms to survive and thrive in an oxygen-rich environment. In order to survive, aerobic organisms have learned to use oxygen to breathe, eliminating the free radicals that can oxidize molecules and damage their bodies. This evolution was made possible by 3 new amino acids integrated into their genome: methionine, tryptophan, and selenocysteine. Since then, all aerobic organisms have learned to survive in the presence of oxygen, provided it remains at normal concentrations. If it exceeds these concentrations, oxygen remains unconditionally poisonous. Although this change of atmosphere was fatal to most life forms of the time, new organisms were emerging in the form of eukaryotes and metazoans. Eukaryotes include all organisms, whether unicellular or multicellular, as long as they contain a nucleus and organelles specialized for respiration. Today, eukaryotes encompass 3 major kingdoms of the living world: plants, fungi, and animals. The second group of organisms to develop is the metazoans. They are multicellular, consisting of a nucleus and organelles, and feed on organic matter. Their cells form tissues. The atmosphere is totally destabilized. Microorganisms have consumed most of the nutrients they needed to form oxygen. But there was another consequence for the climate. During this great oxygenation event, atmospheric oxygen increased considerably, thereby reducing methane. Oxygen combines with methane to form carbon dioxide and water. The latter do not retain heat as well as methane. At the same time, a mass extinction occurred for the majority of life forms, which were then anaerobic. For them, oxygen was toxic. At the same time, volcanic activity slowed for 250 million years. These events combined to produce less carbon dioxide in the atmosphere, resulting in a greatly reduced greenhouse effect. Temperatures fell, and the planet entered an ice age. For the first time, the Earth's surface would have been entirely covered in ice. This abrupt change in climate is linked to the transformation of the atmosphere that occurred with the evolution of life. Photosynthesis processes carried out by prokaryotic bacteria released oxygen. At the same time, the limestone that forms traps CO2, considerably reducing its quantity in the atmosphere. The reduction in greenhouse gas emissions led to a drop in the Earth's temperature. At that time, the sun provided only 85% of the energy it does today. Temperatures reached as low as -50 degrees Celsius, or -58 degrees Fahrenheit, for tens of thousands of years. The water present at the time becomes ice. Albedo increases, accentuating cooling and freeze-up. For millions of years, the Earth resembled an enormous ball of ice, with a surface temperature of -10 degrees Celsius, or 14 degrees Fahrenheit. Scientists believe that this Heronian ice age brought photosynthesis to a virtual standstill. The Heronian glaciation occurred at a very early time in the Earth's history, long before the existence of dinosaurs or most of the life forms we know today. Its probable consequences have been erased by the billions of years of geological evolution that followed. The evidence of this period has been overturned by subsequent geological changes. Nevertheless, scientists have been able to trace the formation of vast glacial deposits. These accumulated in the form of sedimentary rocks, principally tillites, formed by the compaction of continental sediments containing material carried by glaciers, and lamanites. Glacial activity modifies the landscape, forming lakes and valleys. Such a drop in temperature must also have altered ocean circulation and atmospheric composition. Glaciation also had a geochemical influence, modifying the cycle of elements such as iron and oxygen in the oceans. Finally, this change in climate imposed difficult survival conditions on the life forms that remained at the time, and this certainly had an impact on their evolution, as well as on future life forms. And yet, life survives the cold. In principle, below the freezing point of water, the metabolic functions of organisms are interrupted. As water crystallizes, it invades the cytoplasm in organisms. Cell tissues are destroyed, and the organism dies. Yet microbial life can persist in such a seemingly hostile environment, and the return to life following the ice ages is proof of this. When imprisoned in ice, some microbes continue to metabolize at a much lower rate than normal, depending on the temperature and quantity of food available. Others suspend all energy consumption or form spores to survive in the absence of energy. As conditions improve, mechanisms are put in place to repair cellular damage. Microbial life faced with starvation will react in different ways. Some organisms will suffer, while others will react in different ways. They may develop ultra-microcells, synthesize stress proteins, or express starvation genes to become more resistant. Another solution is to reduce metabolism and become dormant, or to form spores. Cold shock and falling temperatures also place different demands on microbes. They will develop methods to counter the loss of membrane fluidity, the reduced rate of enzymatic processes, and cell damage. Ice subjected to extremely low temperatures, such as during glaciations, displays unusual properties. Liquid veins can be found. These are insoluble impurities in the crystalline structure that concentrate in the liquid phase and form a vein-like network, measuring just a few micrometers. These famous veins contain liquid water, nutrients, and biomolecules, a veritable oasis for the metabolism of the microorganisms trapped there. However, microbes must be able to tolerate a pH as low as 0 if they are to benefit from the energy present in acidic ice veins. The acidophiles most frequently found in liquid veins are eukaryotic cells, bacteria, and archaea. There is also a surface of mineral grains dragged along by the ice, which develops a thin film of water that remains in a liquid state. This allows microorganisms to extract nutrients and the energy they need. They will have to adapt to keep their cytoplasm fluid, even when confronted with a temperature of -50 degrees Celsius or 58 degrees Fahrenheit. Despite the catastrophic situation that befell the Earth during the Ice Age, its heart continues to be active. The CO2 emitted by volcanoes accumulates under the ice and is suddenly released into the atmosphere, reinforcing the greenhouse effect and raising surface temperatures. Deglaciation then takes place in just a few tens of thousands of years. Volcanoes have also helped preserve life. They have kept certain areas out of the ice. Thanks to them, many species have been able to survive episodes of glaciation. As soon as the climate becomes milder again, species gradually colonize the regions surrounding these geothermal refuges. Studies carried out in the vicinity of these geothermal zones have shown a richness of biodiversity that diminishes with distance. Hundreds of millions of years have passed since the Heronian glaciation. The Earth has since been rebuilt. 800 million years ago, it consisted of a single vast continent, Rodinia, surrounded by a gigantic ocean, Merovia. At that time, the average temperature was 10.5 degrees Celsius, or 51 degrees Fahrenheit, and the young sun emitted 6% less light than it does today. The day lasted less than 22 hours, and the year comprised 13 months. Present-day North America forms the heart of Rodinia. Australia lies at our latitudes and Antarctica almost at the equator. As a result of magmatic movements and volcanic activity, Rodinia began to fragment around 750 million years ago. Initially, it split into two blocks, in the middle of which a new ocean, Panthalassa, the ancestor of the Pacific Ocean, was born. The part that today represents North America heads towards the ice-covered South Pole. The land in the northern half of Rodinia heads towards the North Pole. Between the two halves lies the Congo Craton. This is a hard, stable zone that was once consolidated to the lithosphere. A few million years later, these three parts will come together again to form a new supercontinent called Pangaea. The collisions associated with this future meeting will give rise to mountains, the Pan-African orogeny. At this time, the mid-oceanic ripples are not very active, and average temperatures are falling. During the Varanger glaciation, the Earth actually underwent two glacial episodes. The first, known as the Sturtian glaciation, began 720 million years ago and ended 660 million years ago. Over the next 10 million years, temperatures rose again, forming an interglacial period. The second glacial episode, known as the Merino glaciation, occurred between 650 and 630 million years ago. These periods of severe glaciation are recognized as the most severe in Earth's history. Since the last ice age, the Earth has seen a resurgence in the diversity of life. There are new types of algae, such as chromophytes, an evolutionary grade grouping organisms with chlorophyll, but also haptophytes, a division of unicellular algae characterized by the presence of a particular extremity called a haptonym, or dinoflagellates, a class of highly diversified unicellular aquatic eukaryotic organisms. There are also hyphae, the filaments that make up mycelium and fungal spores. However, no traces of animals such as hydras or sponges have been found during this period. The parts of ancient Rodinia that reach the poles are not warm. Vast expanses of ice take shape. Soils and oceans freeze over hundreds of meters. Polar caps reach the equator. A feedback loop begins as ice reflects more of the sun's rays than darker oceans. The entire planet freezes over. Temperatures plunge to -50 degrees Celsius, and the thickness of ocean ice reaches over a kilometer. The Earth literally becomes a snowball. The distribution of carbon undergoes profound changes, leading to a reduction in oceanic organic productivity. Photosynthesis is halted for millions of years. Runoff water is no longer an agent of erosion. The atmosphere on Earth is distressing. The sun, although present, shines less brightly. The air is dry, cold, and unbreathable. Snowfall is rare, but powerful winds bring a little dynamism to this desolate land. No liquid water circulates. The situation seems catastrophic for life, and yet, at the bottom of the ocean, life persists in the form of algae and bacteria. And not only does it persist, but multicellular organisms also took off just after this ice age. How was this possible when everything seems so unfavorable to the concentration of nutrients and oxygen needed to sustain life? Scientists have carried out computer simulations to reproduce the snowball effect with an equivalent rate of glaciation. It appears that near the equator there are still refuge zones where the ocean would not have frozen. This is how extremophilic organisms were able to isolate themselves in a closed environment. In the end, evolution continued and even accelerated. Microbial life could even survive and develop in solid ice. Glaciers would have played a local role in oxygenating the oceans. Certain metals, such as iron and manganese, the farther away from the grounding line, the rarer the oxidized forms. The grounding line is the zone between the part where the ice floats and the part where it rests on land. During this ice age, the grounding line may well have been a veritable Eden filled with oxygen. This oxygen came from small air bubbles enclosed in the compacted snow that formed the glaciers. At the grounding lines, these bubbles were released into the liquid ocean as the ice melted and rubbed against the rough bedrock. Even as the ice descends to the equator, volcanism continues, and CO2 and methane are constantly being emitted into the atmosphere. Once the concentration of carbon dioxide reaches 350 times today's level, the greenhouse effect becomes sufficient to reverse the glacial situation and melt the ice. Life can resume. Work carried out by Noah Planavsky in the United States reveals that the erosion of continents during the melting of the ice released large quantities of phosphorus. Its high presence in the ocean would have contributed to the proliferation of phytoplankton and consequently the release of oxygen into the atmosphere. 1 million years, the organisms in the Ediacara fauna resemble jellyfish, arthropods, or leaf-like forms. Some resemble nothing known today. Most of the fossils found at this time do not fit easily into today's phyla. Cells, which until then had reproduced by dividing, were now able to stick together, thanks to a protein called collagen, to form the first multicellular animals. The latter are reduced to their simplest expression, i.e., a simple cluster of cells brought together for the benefits they all derive from it. There are no nervous, vascular, or digestive systems as yet. The sponge is the oldest animal known to have come into existence. Today, this living fossil numbers some 10,000 species. The sponge's body is soft and pierced by numerous tiny holes called pores. It is through these pores that the water passes and is filtered to extract the nutrients the sponge needs. The body is supported by a skeleton composed of limestone or silicon. Sponges have an extraordinary capacity for regeneration. Once a part has been removed, it grows back without being damaged. Sponges have a very wide distribution, having colonized both fresh and brackish marine waters to depths of over 5,000 meters or 16,400 feet, as well as shallow waters, regardless of climate. After a period of calm, the climate became capricious again, and a new glaciation occurred during the Upper Ordovician and Silurian periods, around 460 to 420 million years ago. During the Ordovician period, which began 485 million years ago, the continents were grouped together to form the supercontinent Gondwana. This was formed by the fracturing of Rodinia. The history of our planet is punctuated by the merging and separation of its landmasses under the effect of plate tectonics, and the continents never stop moving. While Gondwana settles at the South Pole, 3 other smaller continents, Laurentia, Siberia, and Baltica, and their islands head north to find a warm home near the equator. Here they experience the vagaries of a tropical climate, a hot sun and violent rainfall. The rain that falls from the sky carries with it carbon dioxide from the atmosphere. Combined with the heat, the rocks are dissolved. Carbon-containing ions are released and carried by runoff into the oceans. Marine animals use them to build their shells and skeletons. When they die, the carbonate material is stored in sediments on the seabed. In the long term, the process of taking carbon from the atmosphere and burying it on the ocean floor considerably reduces the amount of atmospheric CO2. The CO2 released by erupting volcanoes is not enough to compensate for these losses. The greenhouse effect diminishes, and temperatures on Earth begin to fall too. This ice age is not, however, an icy one. We're not talking about a snowball Earth here. It's even likely that in its early stages, it contributed to the development of biodiversity. During this period, the number of marine animal genera increased, known as the Great Ordovician Biodiversification Event, or GOBE. The rapid diversification of plankton favors the development of fauna living on the ocean floor. Cambrian trilobites, inarticulate brachiopods, and eucrinoids gave way to cephalopods, crinoids, and articulate brachiopods. Continental seas are shallow and rich in life, and the first coral reefs appear. Mollusks become common, especially bivalves, gastropods, and nautiloids. Corals appear, colonizing the oceans and building immense reefs like veritable ecosystems. They helped enrich the marine fauna. By the end of the Ordovician period, the oceans were teeming with species, each more diverse and varied than the last. From starfish and sponges to sea scorpions and other extraordinary species of clumsily swimming primitive fish. It was also at this time that the first large predators appeared. Until then, animals had been almost exclusively benthic, staying close to the seabed, but orthocerid cephalopods were the first to swim in open water. Trilobites had better watch out, because the terror of the seas is approaching. Here we see a Cambrian. Sporting a straight cone several meters long, it has tentacles that it can wave from all sides. Its bulging eyes enable it to spot its prey in search of its next meal. With a single stroke of the beak, the carapace of a trilobite breaks in two. From now on, the entire food web becomes more complex. Biodiversity may appreciate the drop in temperature compared with the Cambrian, but it will have trouble coping with the next challenge. The Andes-Saharan glaciation contributed to one of the 5 greatest mass extinctions in Earth's history, known as the Ordovician Silurian extinction. Following this extinction, 70% of species disappeared from the Earth's surface. But what was the origin of this glaciation? Far away, several thousand light-years from Earth, a star exploded in a supernova. The gigantic burst of energy releases gamma rays that reach the Earth at the speed of light. These rays penetrate the atmosphere, altering the ozone layer and darkening the planet. Nitrogen and oxygen are transformed into the toxic gas nitrogen dioxide. The latter engulfs the planet to the point of blocking the passage of sunlight. This theory has little support in the scientific community, as the main obstacle to its acceptance is the difficulty of proving it, as traces of the supernova have long since disappeared. But another cause could have had an impact on the climate of the time. Although solar luminosity was around 4.5% less intense than today during the late Ordovician period, a decrease which under today's climatic conditions would lead to glaciation, the atmospheric concentration of carbon dioxide was considerably higher, reaching levels up to 18 times higher than today. This high CO2 concentration would normally have exerted a powerful greenhouse effect, counterbalancing the lower solar luminosity. Parallel to these factors, the Appalachian orogeny was underway, a geological process that spanned approximately 325 to 260 million years. This phenomenon was the result of the subduction of the oceanic tectonic plate beneath the North American continental plate. This geological process contributed to a massive reduction in atmospheric carbon dioxide levels. The explanation lies in the silicate weathering mechanism, a chemical reaction accelerated by the newly formed mountains, which captures carbon dioxide from the atmosphere and stores it in the Earth's crust. In addition, tectonic plate movements have led to significant changes in the distribution of continents, which may have influenced ocean and atmospheric currents, thus modifying the climate. To this we can add the Earth's orbital variations, which also have the potential to influence climate by altering the incidence of solar radiation on the Earth's surface. Whatever the cause, glaciation is underway. As ice accumulates on the continent, sea levels drop. Shallow marine areas are reduced. The first links in the food chain are affected. resulting in famine for all organisms. What was once an ocean teeming with life is totally devastated. In just over 500 years, a third of the existing fauna had disappeared. Another 100,000 years later, the famine continues. Glaciers sucked water from the ocean. Shoals are drained, drying up. The sea retreats several hundred meters, but once again life will prevail. Beyond a certain depth, organisms can find refuge. Nautiloids and eurypterids seek shelter on the seabed. Other animals adapt to cold water. Such is the case of the astrapus. This small primitive fish of the ostracoderm family, barely larger than a hand, has no fins and moves with the back of its body. To feed, it sucks up small invertebrates living on the seabed or the debris of dead animals. Its skin is highly sensitive to vibrations, enabling it to escape the predators of the time. Astrapus was the first to feature a primitive backbone. This creature, relatively insignificant at first glance, is in fact the first known vertebrate fish. This lineage would later give rise to birds, reptiles, and mammals. Astrapus proved particularly resistant to this new ice age. It follows the retreating ocean. It is a picky eater, feeding on whatever it can find. It also tolerates cold water better than other aquatic creatures. The astrapus adapts to this new ordeal, and soon a new variety of fish is born. These were equipped with the first gills, enabling them to absorb food more easily. It would be another 500,000 years before the planet began to warm up. At last, the glaciers melted. But for the species that had managed to survive the Ice Age, it was a new climatic upheaval. The toll was heavy. Over 70% of species disappeared forever. Thanks to nature's formidable resilience, life will resume and write a new chapter in the history of the Earth. The oceans will be repopulated by astonishing species, and the planet will gradually return to its tropical climate until the next ice age, which began in the Carboniferous period around 320 million years ago. We're in the Devonian over 400 million years ago. This was a pivotal period for vertebrates, but also for vegetation. The Earth is still mostly covered by the immense Panthalassa Ocean. Proto-Gondwana slowly slides northwards. It is set to collide with the young continent of Laurasia, formed a few million years earlier. These two continental blocks would unite at the start of the next period, the Carboniferous, to form a single continent, Pangaea. At this time, the ice caps were absent, and the Earth enjoyed a relatively mild climate, around 25 degrees Celsius, or 77 degrees Fahrenheit. The weather was warm and vegetation thrived and conquered terra firma. The first terrestrial plants, characterized by lichens and mosses, appeared around 500 million years ago. Over millions of years, creeping plants evolved into ferns and lycopods, until during the Carboniferous period, over 350 million years ago, for the first time in Earth's history, they became rich in abundant vegetation. Thanks to warm, humid temperatures, the first insects proliferated. For the time being, they are the only terrestrial inhabitants. There are currently no herbivores. As a result, plants are literally colonizing the Earth. The first forests formed, vegetation diversified and became abundant. Large tree ferns grow alongside lycopods a few meters high. The first broad-leaved plants appear. Thanks to photosynthesis and the resulting oxygen, the Earth's atmosphere is enriched with oxygen, which will not be without consequences. Indeed, the rise in oxygen levels combined with high atmospheric pressure favored the most energy-intensive metabolisms, and large terrestrial vertebrates grew to titanic proportions. One of the best-known examples is the dragonfly, Meganeura, with a wingspan of up to 75 centimeters or 30 inches. There's also the Arthropleura, a distant ancestor of millipedes, which measured up to 1.8 meters or 6 feet in length. Rising oxygen levels also led vegetation to become more resistant to fire. It was also at this time that flowering plants appeared. Increased plant development leads to the absorption of large quantities of atmospheric CO2, which considerably reduces the greenhouse effect. Lignin and cellulose from tree trunks and plant debris accumulate. A polar climate process is triggered. Summers are now too cold for winter snow to melt. The snow accumulates and creates sufficient pressure to form ice. Albedo rises as the ice sheet, an ice sheet covering the mainland, expands. A feedback loop is triggered, and the ice extends further and further. Under the effect of this climate change, plants are forced to slow down their evolution. The high oxygen level triggers fires despite the prevailing humidity. The combination of slower vegetation growth and the occurrence of fires causes a further increase in carbon dioxide in the atmosphere, preventing a snowball effect through greenhouse warming. Over the years, the drop in albedo leads to warmer summers and milder winters, limiting snow depth in areas where glaciers grow. The melting of ice caused by global warming leads to a rise in sea level, which now covers the anoxic zones that used to trap carbon. The Earth now has less surface area to sequester carbon, and a greater proportion of carbon dioxide returns to the atmosphere. The Karoo glaciation came to an end 260 million years ago, bringing the Earth's atmospheric oxygen levels close to those of today. Since then, there has been no ice age until the one we're currently in, the Pliocene-Quaternary, which began around 2.58 million years ago. At the end of the Pliocene, today we are in the last great ice age. It began during the Upper Pliocene some 2.58 million years ago. We are currently experiencing an interglacial period, which is reflected in the retreat of glaciers accompanied by global warming. The current ice age is known as the Pliocene-Quaternary glaciation. Since its beginnings, the Earth has undergone several glacial and interglacial periods. Ice caps advance and retreat on timescales ranging from 40,000 to 100,000 years. In the past, they covered the entire globe. Today, they are limited to Greenland, Antarctica, and a few smaller glaciers, such as the one covering Baffin Island. $2.58 million years ago, the continents were roughly as they are today. Scientists estimate that they have not moved more than 100 kilometers or 62 miles in relation to each other since the beginning of this period. During the previous period, the main change was the connection between North and South America. As a result, the Isthmus of Panama is formed, causing the disappearance of warm equatorial currents and the onset of an Atlantic cooling cycle. The climate gradually became drier and colder. The Greenland ice sheet appears, while the Antarctic ice sheet thickens. The collision between the African plate and Europe continues. The Alps continue to orogenize. Combined with tectonic movement, erosion phenomena, and the trapping of atmospheric CO2 in the form of carbonates continue, reducing the concentration of CO2 in the atmosphere. These phenomena are leading to global climate change. Tropical forests are shrinking, with only a small portion remaining around the equator. Savannas and grasses are now conquering every continent. Temperate forests of deciduous trees are found at mid-latitudes, and conifers and tundra at high latitudes. Antarctica is a veritable frozen desert. The marine and terrestrial faunas, although primitive, are now modern. The junction of North and South America and the formation of the Isthmus of Panama have brought about a great deal of inter-American exchange. A wide variety of animals can now migrate from south to north and from north to south. Mammals, reptiles, amphibians, arthropods, and even flightless birds. Mammals, represented by rodents, mastodons, and opossums, are widespread in North America. We also find Gomphotherium, a now extinct species closely resembling today's elephants. It has 4 almost upright tusks and a short, tapir-like proboscis that falls back on the lower jaw. Sloths, glyptodons, and armadillos migrate northwards across the Isthmus of Panama. Carnivores diversify. They include dogs, bears, and canids. In Eurasia, while the presence of primates diminishes, rodents are widespread. Giant rodent species roam the planet's surface. A giant rodent fossil, Joseph-Fortigacea monesi, was discovered on a beach in Uruguay in 1987. It appears to be the largest and heaviest rodent known today. Its closest relative today is the pacarana, a rodent that weighs between 10 and 15 kilograms, or 20 and 35 pounds, and lives in the tropical rainforests of the Andes. Joseph-Fortigacea monesi, on the other hand, measures up to 3 meters or 10 feet in length and weighs up to a ton. His skull alone measures 57 centimeters or 23 inches. In comparison, it's as tall as a bison. Its morphology is similar to that of a guinea pig, but proportionally the size of a hippopotamus. Its jaw is exceptionally strong. In Africa, primates continue to evolve, and ungulates dominate. It was also at this time that the first humans and giraffes appeared. The number of bovid species increases. For the first time since the Cretaceous, and thanks to the great inter-American exchange, North American species mix with those of the South. Marsupials remain dominant in Australia. Wombats and kangaroos appear. Platypuses appear. As the climate changes, tropical flora is replaced by subtropical forests and grasslands. Western Europe was enriched with trees from the birch, oak, and ash families, as well as conifers further north. But the Earth is affected by a gradual cooling, and vertebrates are not indifferent. Evolution continues, but it's getting poorer. Perissodactyl ungulates decline. Conversely, the large artiodactyl ungulates fared much better, as they were satisfied with a coarse diet and better able to withstand periods of food shortage. Mastodons, these gigantic herbivorous mammals, proliferated, and the first elephants appeared. All these giants were an important source of food, and new larger carnivorous mammals appeared. Most of the fauna that reigned at the end of the Pliocene no longer exists today. the ice ages of the next few thousand years took their toll on many of these animals. Some were able to migrate to milder latitudes, while others were doomed to extinction. Even if winters were no harsher than today, the difference lay in the fact that summer no longer existed. The exact number of glaciations during this period is open to debate. 4 major glaciations have been clearly identified, separated by interglacial periods. These were the Gunns glaciation from 760,000 to 530,000 years ago, the Mindell glaciation from 650,000 to 350,000 years ago, the Risk glaciation from 300,000 to 120,000 years ago, and the Worm glaciation from 80,000 to 10,000 years ago. An important detail in the history of the Earth differentiates this ice age from the others. Man is now at the heart of the glaciations. In fact, it was thanks to these ice ages that the great human adventure began. Climate change means lower sea levels. Far from being a tragedy for mankind, this is actually a door opening for our ancestors from the African continent, Homo erectus, to travel north. In Europe, the cold sets in a little more each winter. Forests wither for lack of water. Oak forests give way to cold, arid steppes. The herds of animals that still roam the plains in search of moss and lichen are a godsend for the human tribes living in Europe. With their rudimentary tools, they catch the young animals and take the adult ones that have already died. As the years go by, the extreme cold sets in, with temperatures dropping to minus 30 degrees Celsius or minus 22 degrees Fahrenheit. Glaciers extend as far as Scotland and the northern Netherlands. The tribes of men, who may have ventured out before, return down in search of warmer lands and temperatures. At this time, fire still eluded them. Ice caps more than 3,000 meters or 10,000 feet thick cover Greenland, North America, and Siberia. All the water used to form these glaciers has caused sea levels to drop by 100 meters. New lands are revealed. The men who populate the African continent now have new prospects. Europe and its herds of reindeer were all the more attractive as the tropical forests had not withstood the heat, and savanna and desert now decorated the landscape. This glaciation lasted for around 80,000 years until an interglacial era began, with the climate warming for around 20,000 years. This cycle has been repeated 4 times to date. It has to be said that the most common state of our planet over the last few million years has been glaciation. In the midst of these glaciations, man has been able to adapt. He protected himself against the cold. Glaciations opened up passages in the sea, and man has continued to travel through glaciations and deglaciations. The event that forever changed its relationship with the climate was the domestication of fire 400,000 years ago. From then on, it penetrated cold regions. They could now set up camp, build shelters and cabins. They can live on permanently frozen ground, their means of heating eternally at hand. After an ice age lasting over 90,000 years, the last great deglaciation began 21,500 years ago and lasted around 10,000 years. At the end of this period, the Earth underwent profound changes. Its average temperature rose by around 4 degrees. The gigantic ice caps that once covered a large part of the north, as far north as Belgium in Europe, melted. Sea levels rose by more than 100 meters. flora was radically transformed and the global water cycle intensified. Over 20,000 years ago, the Earth's climate was stable. Atmospheric CO2 levels were relatively low. It was cold, and the oceans were much lower than they are today. 19,000 years ago, a tiny area in the north began to warm up, while the south remained cold and hardly changed at all. CO2 is still low, and the ice caps are not moving. This evolution of the north is undoubtedly due to orbital changes that brought the Earth closer to the Sun in summer, while the south was plunged into shadow. The continents are concentrated in the north at high latitudes. This is where the ice caps were formed. Even the slightest warming, of the order of a fraction of a degree, is enough to cause a large volume of ice to melt. Fresh water then flows into the Atlantic. The Atlantic is like an ocean corridor, linking the two poles. It carries warm water from the intertropical Atlantic far northwards. Once in the polar zone, this water loses its heat to the atmosphere. It becomes denser and sinks to the depths before flowing southwards. This circulation brings heat from the southern hemisphere to the northern hemisphere. However, if an influx of lighter fresh water occurs in the North Atlantic, the heat in the south may have accumulated as the north cooled. This phenomenon may explain why, 17,500 years ago, the situation shifted. Temperatures in the south rose and the north cooled. Atmospheric CO2 levels rise slowly. After 1,500 years, temperatures in the north also start to rise again, along with the amount of atmospheric CO2. This time, 16,000 years ago, warming affected all latitudes and zones. Atmospheric CO2 levels rose at a faster rate. According to scientific research, warming in the southern hemisphere caused massive degassing of the Southern Ocean, releasing massive amounts of carbon dioxide. By 10,000 years ago, warming had reached its definitive value. Atmospheric carbon dioxide levels stabilized. Over the next 4,000 years, the ice caps will retreat. This scenario could foreshadow the future of our planet if it is subjected to another episode of massive warming. In just 3 centuries, human activity has produced and released into the atmosphere 30% more carbon dioxide than during the last deglaciation, which lasted 10,000 years. For just over 10,000 years, the Earth has been in an interglacial period, characterized as a warm period known as the Holocene. Melting glaciers caused sea levels to rise to their present levels 6,000 years ago. It was at this time that the Black Sea began to fill up. The ice sheets gradually melted, and the land that had been hidden beneath them was freed from the ice and brought to the surface. Temperate fauna and flora reclaimed the middle and high latitudes. As for those that were adapted to the cold climate, they occupy other ecological niches. The rise in temperature during this interglacial period is not without consequences. In tropical zones, rainfall increases, leading to the shrinking of deserts. Rising sea levels isolate the British Isles from the European continent, moving habitable areas northwards. While flora and fauna have not changed significantly, the distribution of species has. Herds of large herbivores left the tropics as they became invaded by vast expanses of forest. They are now interested in the savannas that have appeared in the northern and southern deserts. In the northern hemisphere and Australia, megafauna are becoming less and less present. large mammals are affected by a wave of extinction, the disappearance of mammoths and other large herbivores has had an impact on the climate. When these animals were present 12,000 years ago, they emitted 9.6 million tons of methane per year. As they disappeared, atmospheric methane levels fell, and so did the greenhouse effect, causing the planet to cool. The mammoth was a remarkable animal. Closely resembling today's elephants, it had a large head with a trunk, and a massive body with 4 pillared limbs. The largest species reached between 6 and 8 tons. Throughout their evolution, they adapted to ice ages and cold waves. The size of their ears and tails has diminished considerably. The hair, most exposed to thermal shock, could reach a meter in length. Their wool was 6 times thicker than a human hair. Their highly developed sinuses enabled them to process immense quantities of cold air. Their prominent tusks could reach up to 5 meters or 16 feet, an ideal tool for flushing out grass buried under the snow. Mammoths aren't the only ones affected by the extinction wave. In Australia, large species of reptiles and marsupials are disappearing. In North America, megafauna are not spared either. Mastodons, relatives of mammoths and elephants, are suddenly disappearing. The great land sloths, which grew to the size of cows, did not survive. In the family of large carnivores, the American cheetah, the saber-toothed tiger, and the American lion are no more. Giant armadillos, giant bears, and giant beavers have also disappeared. Man's favorite game, the herbivorous wood-eating mammals such as the antelope, are also disappearing from North America. This megafauna has a long lifespan, a low mortality rate, and a slow population increase. Few predators are capable of attacking their adult specimens. Nevertheless, megafauna are highly vulnerable to climate change. The arrival and spread of early man coincided with the beginning of the extinction wave. The mammoth is an animal that provides mankind with everything it needs: ivory for tools, bones for building huts, fur for covering up and protecting itself from the cold, meat as food, and even grease to keep the fire burning. Man has therefore not hesitated to overhunt this giant of history, contributing to its disappearance. But man and hunting were not the only factors behind the mammoth's disappearance. Mammoths were extremely well adapted to the cold. Global warming during the Holocene caused the evolution of grass and shrub-filled steppes into coniferous forests and tundra regions. Although herbivorous, the mammoth's dentition was probably not equipped to consume tree foliage. Some species, such as the woolly mammoth with its thick fur that kept it warm even in freezing climates, could have died out from hypothermia, while others, such as the bison, have adapted to climate change. The increase in temperature on Earth has not been sustainable for them. Ever since the discovery of the ice ages in the 18th century, mankind has been wondering whether an ice age might return. The Earth is currently in an interglacial period. The periodic and alternating nature of glacial and interglacial periods is now well established, as is the inevitable return of the ice ages. Since its very beginnings, the Earth's climate has been influenced by greenhouse gases and astronomical forcing. The latter is due to the slow changes in the inclination of the Earth's axis and the shape of its orbit. Glaciations in the Northern Hemisphere during the Quaternary era are punctuated by variations in obliquity. The first ice ages, between 3 and 1 million years ago, occurred in regular cycles every 41,000 years. Over the last million years, the periodicity has increased to 100,000 years. The ice caps go from deglaciation to glaciation when astronomical forcing crosses a critical threshold, and vice versa. 420,000 years ago, the interglacial period lasted almost 30,000 years instead of the usual 10,000, because astronomical variations were not strong enough to reverse the system. The Earth is still in this situation today, and it is possible that the interglacial period we are currently in will persist naturally for another 50,000 years if the carbon cycle remains undisturbed. The visual pathway is undeniably one of the wonders of nature. The ability to see near and far, in black and white and in color, while embracing a wide field of vision, certainly represents an evolutionary advantage of remarkable, almost miraculous magnitude. To understand the history of vision, we need to go back in time to explore the theories of eye evolution. Step by step, we understand how this organ has developed over time. Scientists have been divided into two camps. On the one hand, there are those who believe that eyes had a polyphylogenetic origin, meaning that the eye would have evolved independently several times in different species. On the other, those who believed in a monophylogenetic theory, asserting that all eyes derive from a common ancestor, and that each eye type had evolved separately according to the specific needs of the organism. According to recent studies, the trend favors the second theory, i.e., the monophylogenetic origin of eyes. Genetic research has revealed that the PAX6 gene, responsible for light detection, is common to all sighted species. The presence of all three eye types in mollusks intrinsically suggests a monophylogenetic origin of this visual organ. Exactly how long it took for these developments from a single ancestor species to occur remains unknown. Biologists have identified 4 key stages in the evolution of the eye: emergence of efficient photopigments, acquisition of directionality, formation of the photoreceptor membrane fold, and development of optical focusing ability. "600 million years ago, the first organisms developed the most primitive visual organ, consisting of a single photoreceptor, composed of 2 distinct chemical substances, a light-sensitive receptor and a pigment. Its unique function was to detect the presence or absence of light, acting as a kind of electrical switch, with only 2 possible states, on or off. The use of this structure was relatively simple, as it was not used for the perception of movement or localization like our modern eyes. Instead, its role was dedicated to a process called photoperiodicity, which was equivalent to the modern circadian rhythm. In the presence of light, certain reactions occurred, while in its absence they were inhibited. Let's meet a specimen equipped with this very first visual system. This marine animal is very primitive. The hagfish resembles an eel and has no jaws. The hagfish's eyes are very simple and resemble the pineal organs found in non-mammalian vertebrates, especially that they are positioned on either side of its head. Curiously, despite these eyes, the hagfish appears almost blind and reacts very little to light, even when its eyes are removed. It would seem that hagfish eyes aren't really made for vision. In fact, they seem to play a role similar to that of a circadian organ, i.e. they could help it regulate its biological rhythms, like our internal clock. A remarkable example of the evolution of the vision system can be seen in the developmental changes observed in the eyes of lampreys. These changes support the hypothesis that the lamprey's ancestors succeeded those of the hagfish in the course of evolution. Indeed, the Amocoetes, or lamprey larvae, have eye characteristics similar to those of the hagfish. In the larval stage, its eyes are small, sunken under the skin, and have a relatively undifferentiated retina, giving it very limited or non-existent vision. Over time, however, a slow process of neuronal differentiation takes place in a specific order. Ganglion cells, followed by amacrine and horizontal cells, then photoreceptors, and finally bipolar cells are formed. This sequence of differentiation is similar to that observed in jawed vertebrates for the development of their retina. When metamorphosis occurs over hundreds of thousands of years, the lamprey's eye undergoes significant transformations. It enlarges considerably, its retina differentiates completely, the lens develops, the cornea divides into scleral and dermal layers, allowing the eye to move relative to the epidermis. As in jawed fish, extraocular muscles develop, and finally, the eye bursts through the surface to form a visual organ similar to that of vertebrates. The process of eye development in lampreys suggests that these animals have inherited their visual apparatus from a common ancestor they share with hagfish. The existence of a hagfish-like larval eye, which transformed into a vertebrate-like eye in adulthood, supports the idea that lampreys evolved from a common ancestor with hagfish. The next step in the evolution of the eye was the fusion of these photoreceptors to form an eye spot. This advance enabled the organism to detect the intensity of light. In simple terms, if several photoreceptors were activated in an eye spot, this meant that there was strong luminosity and vice versa. This adaptation improved the perception of surrounding brightness. Once again, scientists have discovered clues in embryonic development. Early in the process, a neural structure develops and gives rise to the eye, forming two sacs, or vesicles, on either side. Later, each vesicle coils to form a C-shaped retina, which lines the inside of the eye. It's very likely that evolution followed a similar pattern. At a later stage in embryological development, as the retina folds inwards, the lens forms, leading to a thickening of the outer surface of the embryo, known as the ectoderm. The ectoderm swells into the curved space created by the C-shaped retina, gradually detaching from the rest of the ectoderm to become an independent floating element. It is likely that a similar sequence of changes took place in the course of evolution. The exact date of this change remains unknown, but in 1994, researchers at Lund University in Sweden demonstrated that the optical components of the eye could evolve relatively rapidly, within the space of 1 million years. If this is the case, it is conceivable that the image-forming eye emerged from the non-visual proto-eye in a short period of time on the geological scale. In parallel with the emergence of the eye spot, organisms also developed locomotion mechanisms such as flagella and cilia. These developments led to the need to localize the light source. To fulfill this role, several eye patches fused together to form an eye patch, like a patchwork quilt. These eye patches enabled the organism to roughly locate the light source and orient itself in its direction. By grouping several eye patches together in this way, organisms were able to improve their ability to perceive and move in relation to ambient light. In order to improve localization, the eye patch had to undergo progressive expansion. However, space was an evolutionary constraint. In response to this challenge, nature came up with a solution: the eye cup, also known as the optical cup. The development of the eye cup in vertebrates proceeds as follows. The eyes take shape from evaginations of the developing diencephalon. Depressions known as optic grooves form on either side of the rostral neural plate. As the neural plate folds upwards and inwards to form the neural tube, these growing regions project outwards and are called optic vesicles. When the expanding optic vesicle comes into contact with the surface ectoderm in a specific region called the lens placode, this induces changes in both tissues. The optic vesicle then invaginates to form the optic cup, while the ectoderm undergoes differentiation to form the lens hollow, which later transforms into the lens itself. It is sometimes suggested that the invagination of the optic cup results from the growth of the lens, but observations show that this cannot be the case. In fact, a normal ocular cup and retina can develop in the absence of a lens, indicating that these developmental processes are independent and follow distinct pathways. This adaptation enabled the organism to precisely determine the angle of the incident light beam according to the photoreceptor activated. Thus, the optic cup played an important role in improving the ability to localize the light source, despite the spatial constraints faced by the organism. By this stage, the primitive eye was fully formed, and all the species that possessed this organ entered into fierce competition for available resources. This competition gave rise to a major evolutionary event, known as the Cambrian explosion, which occurred around 540 million years ago. Scientists argue that the evolution of the eye accelerated rapidly during this period, as precise detection of prey and light gave organisms a survival advantage. So the better the eye, the better the organism's ability to survive. This selective competition for improved visual systems played a key role in the rise in diversity and complexity of animal species during the Cambrian explosion. After the Cambrian explosion, the next period saw the evolution of two essential eye structures: the iris, followed by the lens. The first step was to isolate the eye from the outside environment, which was achieved by the development of a transparent epithelial layer, now known as the cornea. Once the light detection system had been separated, the next evolutionary advantage was the ability to focus. Increased focusing power dramatically improved object localization. The first structure to develop this enhanced focusing power was the iris. Thanks to this adaptation, all incoming light was channeled into a single beam, according to the direction of the light rays, greatly improving the accuracy of light source localization. The next major development was the crystalline lens. This lens allowed light rays traveling long distances to focus directly on the retina rather than behind it. This enabled the organism not only to locate the light source precisely, but also to perceive the shape, depth, and other properties of surrounding objects in greater detail. The lens thus opened up new sensory perspectives for organisms with advanced visual systems. With the advent of the lens to capture light and focus images, the eye acquired a greatly enhanced capacity to gather information. This improvement would have generated selective pressures favoring the emergence of more efficient signal processing in the retina, going beyond the simple connections between photoreceptors and output neurons. To meet this requirement, evolution has modified the cell maturation process so that certain developing cells, instead of becoming ciliary photoreceptors, become retinal bipolar cells, which intercalate between the photoreceptor layer and the output neuron layer. This is why retinal bipolar cells bear a strong resemblance to rod and cone cells, although they lack rhodopsin and are not sensitive to light. Instead, they react to the chemical or neurotransmitter released by the photoreceptors, enabling them to play an essential role in processing visual signals before passing them on to the output neurons. Camera-type eyes offer a wide field of vision, usually around 180 degrees. In practice, however, our brains are limited in their ability to process all the visual information available at any one time, due to the limited number of nerve fibers connecting our eyes to our brains. As a result, we can only sample a fraction of the visual data available. It is likely that the first camera-like eyes faced an even more severe limitation, with probably an even smaller number of nerve fibers. This constraint would have exerted considerable selective pressure in favor of the evolution of muscles to move the eyes. The presence of such muscles must have existed around 500 million years ago, as the arrangement of these muscles in lampreys, whose lineage goes back that far, is almost identical to that of jawed vertebrates, including us humans. Despite the ingenious features of vertebrate eye evolution, a number of rather counterproductive traits can be identified. For example, the retina is positioned upside down, which means that light has to pass through the entire thickness of the retina, including nerve fibers and intermediate cell bodies that scatter light and impair image quality before reaching the light-sensitive photoreceptors. In addition, blood vessels cover the inner surface of the retina, casting unwanted shadows on the photoreceptor layer. Another tricky aspect is the presence of a blind spot, where nerve fibers that cross the surface of the retina come together before tunneling through it to emerge as the optic nerve at the back of the eye. This list of inelegant features could be extended to include other examples. These imperfections are by no means inevitable features of a camera eye, as octopuses and squids have evolved independently of camera eyes which do not have these defects. Examining the vertebrate eye in an evolutionary context highlights these seemingly absurd shortcomings as the consequences of an ancient sequence of evolutionary steps, each of which brought advantages to our vertebrate ancestors, long before they developed advanced vision. The eye has evolved in many different ways over time, giving rise to a variety of features in different species. Jellyfish, for example, have eyes, and although they have no brain, these eyes transmit information directly to the muscles for rapid reaction. Nautiluses have an aperture without a crystalline lens, adopting a different focusing mechanism. Flies, on the other hand, have many eyes, giving them a wider field of vision. The diversity of ocular adaptations continues with predators, which have forward-facing eyes for better depth perception, while prey have lateral eyes, offering a wide field of vision for detecting potential dangers. In the case of humans, they have developed the ability to artificially correct their vision using devices such as spectacles, enabling them to compensate for certain visual imperfections. Ultimately, the list of ocular adaptations is truly infinite, with each species evolving uniquely to suit its environment and specific needs. All the auditory organs of today's terrestrial vertebrates, whether reptiles, birds, or mammals, have evolved in completely distinct ways. Ciliated cells, the sensory cells at the base of the auditory organs of all vertebrates, emerged very early in the evolutionary history of animals. They play a significant role in today's non-vertebrate animals, such as ascidians, and can be traced back to the first animals with well-developed tissues, the cnidarians, like sea anemones. The auditory part of the ear evolved from a vestibular organ whose evolutionary origin, in particular the sensory cells themselves, resembles that of the lateral line, a body surface organ used by fish to detect the movement of fluids in water. The lateral line has sensory hair cells similar to those in the cochlea, whose responses are strongly influenced by viscous damping and are linked by a gel-like structure. The damping problem was solved early on by evolving the active mechanisms of the sensory cells and linking them via a gel-like structure to increase both their sensitivity and frequency selectivity. These two features have survived 500 million years of evolution and are common to vertebrate inner ears. During the Carboniferous period, from -359 to -299 million years ago, tetrapod ancestors successfully migrated to terrestrial life, paving the way for further adaptations of existing sensory systems, including hearing, from aquatic to terrestrial life. Thus, over hundreds of millions of years, progressive adjustments were made to these organisms so that they could adapt to higher frequency stimuli, first in an aquatic environment and then in an aerial one. It is assumed that the ancestors of amniote vertebrates had a limited hearing capacity for loud, low-frequency sounds, as they lacked a tympanic middle ear. The first terrestrial vertebrates had an auditory papilla that rested on solid tissue and was covered by an otolith, a small mineral structure present in the inner ear of certain animals, notably fish and reptiles, responsible for detecting movement and balance, and composed of calcium carbonate crystals. In amphibians, the otolith membrane was replaced by a tectorial lining that contained no otoliths. In all subsequent lineages, the basilar membrane appeared, freely suspended. In other lines, the basilar membrane showed some adaptation, with moderate frequency selectivity. Around 100 million years after the separation of the tetrapod lineages during the Triassic period, at least 5 independent developments of tympanic middle ears were observed. These developments functioned as essential impedance matching mechanisms for the efficient detection of airborne sounds. The absence of an effective middle ear prevents the detection of high-frequency sounds. This gave rise to parallel evolutions, leading to major differences in the auditory systems of tetrapod vertebrates between different clades, while ultimately fulfilling the same function: detecting, transmitting, transducing, and encoding airborne sounds. Paleontological, morphological, and developmental evidence suggests that lepidosaurs, such as lizards and snakes, as well as archosaurs, which include birds and crocodiles, independently developed a middle ear, composed of a single ossicle. Firstly, in birds and crocodilians, The structure of the auditory organ shows marked similarities, such as the presence and distribution of similar cell types. It is therefore reasonable to assume that this structural configuration was reached or initiated at the latest around the separation of their ancestral lineages some 160 million years ago. Similar reasoning applies to lepidosauurs. In Tuataris, which separated early from other lepidosaur lineages, the cellular configuration of the auditory papilla is similar than in lizards. In Tuataras, as in turtles, there is only one type of cylated cell, and all cylated cells have the same orientation. Thus, the more complex structure typically seen in lizards seems to have arisen after the Tuatara lineage separated from the squamates. However, it is not known whether lepidosaurs, including the Tuataris, developed a tympanic middle ear all at once. Finally, in mammals, the characteristic structure of the auditory organ, known as the organ of Corti, is essentially found in all recent groups of mammals, although there are marked differences between monotremes and therian mammals. We can therefore assume that the organ of Corti appeared before these lineages split, between 220 and 150 million years ago. This comparative analysis suggests that the evolution of the tympanic middle ear in each lineage had significant implications for the later development of the auditory organs of the inner ear. The reduction in bone size and the emergence of new configurations to form the tympanic middle ear probably improved not only auditory transmission in general, but especially that of higher frequencies. This new feature undoubtedly exerted additional selection pressures on the auditory organ to enable optimal transduction of information and its transmission to the brain. Over time, the mammalian cochlea evolved specifically in different subgroups. Over the course of several subsequent geological periods, mammals diverged considerably, and although the basic structure of their cochlea was relatively constant, differences in length and various specializations emerged. Not only did cochlea develop huge differences in length, they also developed specific specializations in groups pursuing different strategies for locating prey, using ultrasonic frequencies in the air, such as bats, and high ultrasonic frequencies in the water, such as toothed whales. For example, some bat species have developed cochlear regions where the length of the organ of Corti, dedicated to specific narrow frequency ranges, is considerably enlarged. These adaptations have enabled bats to better detect and localize sounds in their environment, improving their hunting and navigational abilities. On the other hand, there is no reason to expect mammalian auditory organs to be necessarily superior or more sophisticated than those of other groups. Each present-day vertebrate group has developed an appropriate auditory solution according to the evolutionary pressures dictated by the environment, predator-prey relationships, and intraspecific communication needs. Acute sensitivity and precision in frequency resolution, characteristics commonly associated with mammalian auditory organs, have evolved independently in all groups of modern terrestrial vertebrates, in response to their own specific evolution and adaptation. Indeed, some species of lizard possess auditory nerve fibers tuned to some of the finest frequencies of any vertebrate, including most mammals. Birds of prey, particularly owls such as the barn owl, rely almost exclusively on hearing to locate their prey, so auditory sensitivity exceeds that of most mammalian species. Echolocating mammals such as bats and dolphins exhibit a wide range of hearing frequencies, a characteristic almost equaled by certain species of frog, which use ultrasonic calls to communicate. In humans, although generation times are 10 times longer than in fish, the issue is not so much the creation of hundreds of new species, but rather the adaptation of a sensory organ to optimize the processing of communication signals vital for survival and hence reproduction. Whether the evolution of the human cochlea paralleled the development of language is a fascinating question that could be explored by future research. All living cells are irritable to certain chemicals, and this predisposition of cells to be perturbed by chemicals is likely to have led to the eventual evolution of specific receptor proteins to detect chemical signals, and ultimately to specific chemosensory organs and systems, such as the sense of smell. Olfactory capacity, used for chemical communication, is widespread in the majority of living beings belonging to the animal kingdom, enabling the perception and identification of chemical signals. The sensitivity and chemical range of animals' olfactory systems are remarkable, giving them the ability to detect and distinguish between thousands of different odor molecules. In the animal world, olfaction has evolved to enable the detection of signals from the chemical environment, which contains decisive information on the direction to follow, food sources, the right time to reproduce, as well as stimuli to consider pleasant or dangerous. An animal's reaction to chemical signals is the result of a combination of mechanisms acquired during its lifetime and innate predispositions resulting from the cumulative impact of individual experience and evolution through the generations on the olfactory nervous system. Evolution has shaped the olfactory system in response to two challenges: the need to explore a wide range of chemical information and the specific sensory challenges faced by each animal species. Generally speaking, vertebrates develop an olfactory system that detects odorants and pheromones through their interaction with cell surface receptors specialized on olfactory sensory neurons and organized in dramatic and subtle ways, enabling adaptations in response to major changes in environment and lifestyle, such as the transition from ocean to land and the predominant development of vision. In terrestrial vertebrates and insects, we often think of olfaction as a chemosensory modality dedicated to the detection of low concentrations of airborne volatile chemicals. Yet aquatic fish and crustaceans, while not encountering airborne volatile odorants, possess sensory systems anatomically similar to the olfactory systems of terrestrial animals. For these aquatic animals, odors are sapid molecules in solution. Olfaction is therefore not necessarily the detection of volatile molecules in the air. In this context, it has been suggested that during the transition between aquatic and terrestrial environments, new demands emerge for chemosensory systems linked to odor detection. This happened in a remarkable way, as olfactory stimuli evolved from hydrophilic to predominantly hydrophobic and volatile compounds. This transition granted terrestrial vertebrates an evolutionary option to detect volatile water-insoluble odorants. As a result, it is thought that the complex biosynthetic pathways required to produce volatile compounds, as well as the highly complex combinatorial coding that characterizes olfactory perceptions, driven by olfactory receptors, emerged suddenly in just a few 100,000 years. Comparative genomic and phylogenetic studies argue that chemosensory receptors were conserved across aquatic and terrestrial taxa, and that mammalian-like olfactory receptor genes evolved in marine environments before the first vertebrates appeared. These changes in olfactory receptors at protein level, altering their interaction with odorant molecules, play a key role in the evolution of olfactory perception. However, researchers have discovered that this adaptive evolution of olfactory perception can also be influenced by co-expressed olfactory receptors undergoing changes in copy number and protein sequence. Selection also acts on individual olfactory receptor families or subfamilies, favoring specialized adaptation or facilitating chemical communication between members of the same species. Finally, genetic variation among the thousands of unique olfactory receptor genes can lead to behavioral modifications in response to specific chemical signals. Thus, the combination of these mechanisms enables vertebrates to adapt in precise and complex ways to their changing chemical environment. An animal's genome evolves in such a way as to be closely linked to the specific tasks resolved by its nervous system, enabling it to detect odors and respond appropriately to promote survival and reproduction. Recent studies have revealed the presence of ancestral versions of synaptic genes involved in fundamental neuronal communication in various organisms without a nervous system, suggesting a progressive evolution of olfaction. In parallel, studies have clarified that the olfactory capacities of multicellular organisms, as well as chemosensory learning abilities, evolve from life forms not equipped with complex neural networks. In particular, prokaryotes have been proposed as model systems for understanding the evolution of olfactory systems in higher organisms. In fact, bacteria respond to volatile chemical gradients, but also use contact-dependent signaling. In addition, a unicellular but multinucleate eukaryotic organism, Viscerum polycephalum, has been shown to be capable of chemosensory detection. Studies have shown that olfactory receptor cells are primary bipolar neurons with a similar morphology between species. They have filamentous dendrites to increase the surface area for stimulus capture, and an axon that extends to the central nervous system. Dendritic processes are mainly of ciliary origin in most animals. Although the overall morphology is conserved, the fine details of olfactory receptor cell morphology can be adaptive, i.e., they can vary in a habitat-dependent rather than species-dependent manner. For example, the olfactory receptor cells of land-dwelling crustaceans, such as the giant thief crab, have a morphology closer to that of insects than to that of marine crustaceans. Crustaceans share a common ancestor with insects, and as they lack olfactory receptors, it has been proposed that these receptors evolved when prehistoric insects left the sea to live on land. According to this idea, olfactory receptors evolved because these ancestors must have been able to detect odorous molecules floating in the air, rather than dissolved in water. But a recent discovery contradicts this hypothesis. The discovery of a new type of olfactory receptor, ionotropic receptors, IR, has changed the order of things and opened up new avenues of inquiry into the evolution of olfactory receptors throughout the history of life. These new olfactory receptors are present in crustaceans, in which no classical olfactory receptor had been found until now. Since crustaceans were the first to appear in the history of life, before insects and before vertebrates, we can assume that these IR were inherited long before the appearance of the first insects. During development, the olfactory system takes shape from the olfactory placodes, specialized regions of the anterior ectoderm that share cellular and molecular characteristics with other placodes involved in the development of various cranial senses. Certain cell types of the olfactory system preceded the appearance of vertebrates, as did certain mechanisms involved in placode formation. It is likely that these two elements were already interconnected in the common ancestor of vertebrates and tunicates. In vertebrate ancestors, this system evolved into a more complex organ system, integrating additional tissues and specific morphogenetic processes to develop distinct olfactory components. Subsequently, the ancestral placode divided to give rise to the paired olfactory organs characteristic of most modern vertebrates. Thus, a major olfactory adaptation occurred along the amphibian and terrestrial branches of the vertebrate tree, resulting in the formation of distinct neuroanatomical structures expressing more recently derived olfactory receptors. Aquatic vertebrates such as fish and amphibians have an olfactory system similar to that of terrestrial vertebrates, but specialize for the detection of chemicals present in water rather than air. Insects, on the other hand, have a well-defined system in their antennae for detecting airborne chemicals, generally referred to as the olfactory system. However, this system is evolving convergently, independently of vertebrates, to perform this function. Unlike fish, amphibians have developed a separate vomeronasal organ, which specifically detects attractive sex pheromones specific to amphibians. In addition, they possess an air-filled main olfactory epithelium, which expresses a vast repertoire of novel Class II olfactory receptors that have acquired a role in innate reproductive behaviors. These observations suggest that odor interpretation depends not only on the odorant molecules detected, or ligands, but also on the developmental identities and specialized sensory pathways of the cells that detect these odors. Recent evidence shows that individual mammalian olfactory receptor cells can be activated by several different odorants, and that individual odorants activate several receptor cells expressing different receptor proteins. This combinatorial coding is also present in insects and fish, suggesting that it has been conserved in the evolution of olfaction. Consequently, there are striking similarities between species in the organization of the olfactory pathway, from the nature of olfactory receptor proteins to the organization of the olfactory central nervous system. Although there is a striking evolutionary convergence towards a conserved organization of signaling pathways in vertebrate and invertebrate olfactory systems, the receptor gene families involved have evolved independently. These common organizational features span a wide phylogenetic range of animals. Such conservation also implies that there is an optimal solution to the problem of odor detection and discrimination. Either this solution evolved relatively early and was then retained in evolution, or more likely, animals convergently developed identical or similar solutions to the odor detection and recognition problem. The sense of taste is a specialized chemosensory system, one of the main mechanisms used by animals to assess the nutritional quality of food, helping them to make decisions about consuming beneficial foods while avoiding harmful substances in their daily lives. Unlike touch, vision, hearing, or olfaction, which are called upon in different behavioral situations, the sense of taste has evolved to play a primordial role in food regulation and motivation. Taste systems detect essential nutrients, as well as harmful substances in food, and trigger innate behaviors that lead to the acceptance or rejection of potential food sources. Most vertebrates can perceive 5 basic taste modalities: bitter, umami, sweet, salty, and acidic. All vertebrates have some ability to avoid harmful chemicals and seek out nutrients. It is thought that each sense of taste has evolved to detect and motivate the consumption of essential nutrients, and also to detect and avoid potential poisons. It is widely accepted that sweet taste evolved in plant-eating animals to detect energy-rich simple sugars, such as glucose, fructose, and sucrose. Bitter taste, on the other hand, probably functions to ensure that an animal avoids poisons. Indeed, most poisons are bitter, and most bitter substances are harmful, although this relationship is not perfect. The salty taste allows the detection of sodium, an absolutely essential mineral. When certain species of animal lack sodium, and this usually occurs in herbivorous animals, a powerful appetite for salty taste is aroused. And for many species, salt is consumed even when there is no apparent need. As for the sour taste, many have suggested that it is involved in detecting fruit ripeness. Finally, the 5th basic taste, umami, probably serves to signal amino acids and proteins. By comparing animals from different branches of the evolutionary tree, researchers have deduced that the sense of taste probably evolved over 500 million years ago. This would have occurred before the divergence of terrestrial vertebrates, bony fish, sharks, and lampreys, when their common ancestor, a primitive fish, developed a new type of cell. These chemosensory taste receptor cells, located in taste buds, are able to detect chemical stimuli and send signals to the brain via the taste nerves to generate taste sensations. Taste buds are the gustatory end organs in vertebrates ranging from lampreys to mammals. These end organs respond to a variety of sapid chemicals and transmit signals to afferent nerve fibers from 3 cranial ganglia: facial, glossopharyngeal, and vagus. Over time, taste buds have undergone several modifications to adapt to the dietary needs of different animal species, which explains their varied location in these animals. Not only are they found in the mouth and pharynx, but they can also be widely distributed across the surface of their bodies. Evidence suggests that umami receptors were the first to develop. Researchers have reported the discovery of genes similar to those encoding the receptors used by humans to detect the amino acid glutamate in the genome of the elephant shark, a species that split off from other fish 400 million years ago. Sharks lack bitter taste receptors, suggesting that these genes evolved more recently. Ancient fish were probably the first vertebrate animals capable of detecting sour taste. It is likely that these fish did not taste food with their mouths, but rather could detect acidity in the ocean using the outside of their bodies. Variations in the carbon dioxide dissolved in water can create different levels of acidity, which can be dangerous for fish. So the ability to sense acidity would have been essential for their survival. Bitter taste is perhaps the taste that most intrigues evolutionary biologists. It's the Darwin's finch of taste, engineered and customized to suit a species' ecological niche. Humans have 24 or 25 different bitter receptors, depending on the individual, each recognizing unique combinations of chemicals. Toxic bitter compounds come in all shapes and sizes, so it makes sense that the receptors that recognize them are diverse. The evolution of bitter taste, therefore, also involves distinguishing between different chemicals. The diets of humans and their ancestors are mainly based on plants, which are an abundant and accessible source of nutrition. However, plants have developed physical, reproductive, and chemical defenses to protect themselves from herbivores. Herbivores must therefore contend with various obstacles such as thorns, shells, and symbioses with defending animals. Herbivores have developed various strategies to overcome the toxic defense mechanisms of plants. Among these, a particularly effective and widespread method in vertebrate animals is the detection of bitter flavors. It has long been widely recognized that many plant toxins are perceived as bitter, creating a powerful mechanism for minimizing exposure to these harmful substances. By signaling the presence of toxins in foods before they are fully consumed, bitter taste detection helps avoid their ingestion. This ability plays an essential role in the survival of herbivores, helping them to identify and avoid potentially dangerous plants. The ubiquity of bitter perception in vertebrates and its complex role in humans have raised questions about its evolution. One of the most remarkable revelations of vertebrate comparisons is that bitter taste receptors are a relatively recent evolutionary innovation. While they are present in bony fish, lobe-finned fish, and tetrapods, they are not found in cartilaginous fish. Thus, bitter taste receptors must have first appeared in an aquatic species around 430 million years ago. This makes them the most recent of the chemosensory receptor families in vertebrates. The origins of the receptors involved in taste perception in animals are the subject of much speculation. Similarities between the evolution of vascular plants and the Cambrian explosion in animals suggest that the origin of these receptors may be linked to an important transition in plant to herbivore interactions. However, There is also evidence that these receptors play other roles, such as detecting nutrients in the gut, suggesting that their function as taste receptors may have emerged later. The bitter taste receptor family has diversified within and between taxa, with repertoires of bitter taste receptor genes varying in size across species genomes. Studies show that selective pressures on taste perception represent the most obvious potential driver of diversification of these genes. Fish have different numbers of genes, ranging from 1 in Amazon molly to 74 in coelacanth, but most fish have an intermediate number of genes. Variation in the number of bitter taste receptor genes occurs in different groups of animals, such as fish, amphibians, and saurians. Caecilian amphibians, which include limbless amphibians, have the lowest number of genes, while anurans, which include frogs and toads, have the highest number among all recorded vertebrates, probably reflecting their adaptation to variable lifestyles and environments. In fact, most frogs and toads inhabit both aquatic and terrestrial niches, which necessarily contain a greater variety of toxic substances. Consequently, their ecological needs should encompass very different requirements for their gustatory systems. For example, aquatic factors such as pH have been shown to influence the divergence of taste receptor genes. Most frogs feed on worms, insects, and other small arthropods, which contain more potentially toxic substances. It is known that insects often sequester toxic compounds from plants for their own defense, suggesting that anurans may benefit from the ability to detect dangerous prey. While caecilians are animals adapted to life underground, living in soil layers rich in organic matter, they build tunnels to get around. All caecilians are carnivores, feeding on earthworms, plathelminths, arthropods, frog eggs, and tadpoles. Compared with frogs, caecilians are less sensitive to bitter taste, as animal tissues contain fewer toxic substances. Consequently, we hypothesize that the lifestyle, ecological, and dietary complexity of amphibians raise their evolutionary pressure for a wide variety of taste receptor genes. Saurians, which include crocodiles, lizards, snakes, and birds, also show variation in the number of these genes. Snakes have low numbers, while crocodiles, turtles, and lizards have higher numbers, similar to many fish. Repertoires of bitter taste receptor genes in birds and mammals show great diversity. Penguins and cetaceans have few or no functional genes, while birds such as zebrafinch and white-throated sparrow have several. Primates, including man's closest relatives, have an average number of genes, with the gibbon having the fewest and the chimpanzee the most. Although toxins are a threat to all herbivorous animals, not all species are equally exposed. This variability is reflected in the size of genetic repertoires, which is related to the proportion of plant-based diets for each species. This trend is particularly marked with herbivores having larger repertoires than carnivores. The most extreme example of this variation is found in obligate carnivores such as whales, penguins, and pinnipeds, which swallow their food without chewing, resulting in a total absence of certain genes linked to toxic detection, around 7 to 8 million years ago, humans had one last common ancestor with other great apes. If we look at the current eating habits of great apes, we can deduce the diet of our common ancestor. It seems that this species was omnivorous, feeding mainly on tropical fruits, and also included leaves and insects in its diet. Chimpanzees, our closest relatives, rely heavily on fruit for their caloric intake, with a small proportion of their diet coming from animals such as other apes and insects. Between 4.4 and 2.3 million years ago, human ancestors left the forest for the savanna and other ecosystems, and their food repertoire expanded considerably. Thus, over time, early hominids gradually abandoned the eating habits of forest monkeys and increased their ability to obtain a variety of nutrients in order to adopt a more diversified outdoor diet. Despite this dietary evolution, our need for fruit and our attraction to sugars and acids, shared with other great apes, remained intact. From a nutritional point of view, fruit is attractive because of its sugar content, which provides intrinsic satisfaction, as well as vitamin C, which is essential for life support in hominoids. The question of why humans are sensitive to acidic flavors and even inclined to prefer acidity has sparked debate in the scientific community. Signals from acidic substances do not provide much nutritional value, with a notable exception of vitamin C. However, this exception is significant because, unlike most mammals, monkeys and apes are unable to synthesize vitamin C due to the loss of a functional version of the gene encoding gluconolactone oxidase. The common ancestor of arthropods that suffered this enzymatic loss probably had to rely on a sufficiently rich source of ascorbic acid from fruits and other plants for the enzyme to become useless. It is plausible that the acidic taste played a necessary role as a guide to identifying vitamin C-rich fruit. In addition, the combination of acids with sugars could also have been used to assess the degree of ripeness of the fruit, using mixtures of sweet and sour flavors. Thus, acids are not stimuli for which we have evolved to react in isolation, but rather elements we have experienced in the context of the sugars present in fruit. As a result, sweet and sour flavors are perceived as synergistic in the fruit aroma palette. In addition, acid and sour taste act as indicators of fermentation processes which human beings around the world seek out and consume. Wild apes depend mainly on forest plants, especially young leaves, for most of their daily protein intake. Nevertheless, early hominids and modern humans have shown a preference for consuming slightly more digestible protein sources, such as meat. Although umami taste is not immediately apparent in fresh meats, it develops significantly in aged or cooked meats. Consequently, it seems that chimpanzees do not possess a gustatory system specifically dedicated to detecting the taste of glutamate or ribonucleotides, even though they can perceive these stimuli. The characteristic umami taste of hydrolyzed protein is mainly carried by glutamate and ribonucleotides. Humans have developed a preference for glutamate, ribonucleotides, and umami taste, probably as signals indicating more easily digestible proteins in lightly aged or cooked meats. Indeed, There is ample fossil evidence to suggest that culinary practices predate the advent of modern man. Humans the world over take advantage of a wide variety of plant and animal products that undergo fermentation. Our strong attraction to the taste of free amino acids and ribonucleotides may stem from a tendency to consume fermented foods, including lightly aged and/or cooked meats. This category of food would bring various benefits to the survival of our species. Fermentation facilitates access to macro and micronutrients while providing probiotic bacteria. Although it has been assumed that the pleasant taste of glutamate and ribonucleotides acts as an indicator of protein, many protein-rich foods are not particularly tasty or umami when fresh. It's the process of fermenting or aging these foods that releases the umami, savory taste of protein. Thus, our attraction to amino acids, particularly glutamate, as well as salty taste, may have emerged from our desire for fermented foods, taking advantage of the nutritional improvements and benefits of probiotic bacteria for our species. Humans stand out from other mammals thanks to a unique phenomenon, a large-scale polymorphism of the salivary amylase gene. This change in our genetics could be the result of our past history linked to the consumption of starch-rich tubers, and could have occurred around 100,000 to 200,000 years ago. The number of copies of the salivary amylase gene present in our genome directly influences the production of amylase in our saliva. This enzyme in high quantities and efficiency has the ability to rapidly transform a cooked starch, such as a thick pudding, from a semi-solid to a liquid state in a matter of moments in our mouths. The resulting sugar concentrations can produce sufficient substances such as maltose, which activate brain regions linked to taste and reward. In short, it seems that the human mouth harbors a taste receptor that responds to glucose polymers without necessarily inducing a conscious taste sensation. This receptor can be stimulated by the products of starch decomposition in the mouth, especially in the presence of high levels of salivary amylase. Human beings have a natural tendency to seek out the taste of salt in food. We find the taste of moderate concentrations of salt very appealing, as do other omnivores. But high levels of salt are considered unpalatable to us, as they disrupt the osmotic balance of our body fluids. Salt is a widely used ingredient in food preparation throughout the world, and individuals from different cultures consume roughly the same amount of salt every day. This similarity in salt consumption across cultures suggests that our salt intake is regulated by intrinsic biological factors. A carnivorous animal absorbs salt at every meal, while a herbivore can easily run out of sodium, triggering a desire for salt, which translates into a search for natural licks. An omnivore's exposure to dietary salt would therefore logically be intermediate between that of carnivores and herbivores. It should be noted, however, that humans lose salt through perspiration, which may explain why we have a slightly higher salt intake preference than other omnivores. Since our earliest prehistoric ancestors, the sense of touch has been central to our survival and reproduction. What we touch can have a direct impact on our ability to survive and reproduce. It may be a predator, prey, or parasite, a parent, lover or child, shelter or danger, a tool or food source, the ground beneath our feet, or the branch we use to swing. It's also the first sense to develop, with some fetuses responding to touch in the womb after just 8 weeks. Evolution has created a tactile system made up of different sensors and receptors, which together detect the qualities of what touches our body and what our body touches, such as weight, temperature, and texture, so that we can deduce useful information. Many animals, including some coelantherates, annelid worms, insects, and many other arthropods, birds, and mammals, have hairs or capillary projections that are richly supplied with nerves and serve to indicate to the animal that it is in contact with an object. These hairs may have been specially modified, for example, vibrissae or whiskers in certain areas of the body, such as the face or toes, to provide more sensitive discrimination between stimuli. A second type of tactile receptor, the subcutaneous receptor, is found in the skin. Receptors of this type are found in almost all animals and may consist of free nerve endings or complex corpuscles. Unicellular organisms already possess the capacity for directional detection of tactile stimuli, as can be seen in ciliates such as Paramecia, which move away from the stimulus source following mechanical disruption of their outer membrane. Sponges, the simplest multicellular animals, do not possess neurons, but they do have the capacity to respond to variations in water flow and pressure in response to the movement of their immobile cilia. However, the evolution of neural conduction has led to a radical change in the ability to respond rapidly and flexibly to tactile stimuli. Cnidarians such as jellyfish and hydra, despite their relatively simple nervous systems, can exhibit coordinated patterns of motor response to sensory stimuli, and many have a rich capacity to respond to touch. For example, hydra nematocytes are structures that provide a good model for understanding the mechanoreceptors of more complex invertebrates. In jellyfish, groups of hair cells called tactile combs regulate complex behaviors, including escape, feeding, and locomotion. The advantages of sensitivity to mechanical stimuli provided by hair-like structures may have encouraged their convergent evolution in several animal lineages. For example, the ciliated cells of jellyfish appear to be sufficiently different from those of vertebrates that a common origin for both is unlikely. Studies into the molecular basis of mechanosensation in different classes of animals also suggest that the cellular mechanisms required for tactile detection may have evolved several times. All animals with a central nervous system respond to touch. The nematode, Sanor habditus elegans, is an important model organism in neuroscience. It is the first animal system in which the entire neural network involved in tactile behavior has been identified. This worm has mechanosensory cells located in the integument beneath the cuticle, linked to 6 sensory neurons, 10 interneurons, and 69 motor neurons. In larger, probably more advanced animals, touch-induced behavior becomes more versatile and complex. The leech has developed a central nervous system with a succession of ganglia for each segment. Each ganglion houses 3 groups of mechanosensory neurons, all of which respond to mechanical stimulation of the body, but with different response thresholds. Tactile cells are the most reactive and respond to the gentle touch of the body wall. Pressure cells respond to more intense tactile stimuli, while nociceptive cells are sensitive to very powerful and potentially harmful stimuli. In arthropods, mechanoreceptors are located in strategic areas of the body where movement is most likely to occur. In proprioception, these are the joints, muscles, and tendons. In contact, they are the surface areas where contact with external objects is most likely. Arthropods exploit these strategically important locations in two ways. On the one hand, they use tufts of hairs called capillary fields, or capillary plates, to detect movement between adjacent body segments through proprioception. On the other hand, to detect contact, they use similar hairs but located in different parts of the body, with a particularly high concentration where contact is most likely and/or most important to detect. Spiders and insects also use hair sensilla for proprioception and consequently for active touch detection, where the active movement of a limb must be monitored. Among today's arthropods, only myriapods, crustaceans, and insects have specialized touch sensory structures, the so-called antennae. These sensory organs are equipped with a particularly large number of sensillae, displaying a density far greater than that found on most other body regions. Crustaceans have 2 pairs of antennae, while insects have only 1 pair. The generally accepted hypothesis is that over the course of evolution, insects are likely to have lost their second pair of antennae, meaning that the insect antenna shares a common evolutionary origin with that of crustaceans. Fish and amphibians have mechanoreceptive organs called neuromasts, which are arranged in rows on their body, head, and tail. These neuromasts may be present autonomously on their skin, or inside fluid-filled channels. The main sensory component of each neuromast is a group of hair cells that respond to hydrodynamic stimuli caused by water movement or variations in water pressure. In this way, the lateral lines provide a sophisticated sensory system for detecting the distant origins of water movement, such as predator or prey behavior, water currents, and the topographical features of the underwater environment. Mammalian skin evolved from the integument of earlier vertebrates in a complex evolutionary process that is still only partially identified. What is clear is that the first mammals, or perhaps their reptilian, therapsid ancestors, redeveloped hair as an adaptation of the outer epidermal layers of their integument. While dense hair clearly has a thermoregulatory function, the evolution of hair in mammals cannot be attributed solely to the goal of regulating body temperature. On the contrary, it is very likely that the first hairs were primarily used for tactile functions, and their subsequent evolution gradually gave them a secondary role as insulators. The main sensory function of hairs persists in vibrissae, also known as tactile hairs, present in all Therian mammals, such as marsupials and placentals, with the exception of human beings. In cases such as the naked mole rat, a species that has lost its entire coat, tactile hairs have been maintained throughout the body because of their importance to the sense of touch. Many mammals have evolved to have areas of hairless skin, also known as glabrous skin. In humans, these areas include the skin of the lips, hands, fingertips, and soles of the feet. These are the parts of the body that play a key role in physical interactions with the outside world, and where tactile precision is particularly important. So it's not surprising that hairless skin is characterized by a high density of mechanosensory receptors that are extremely sensitive to delicate touch, which could contribute to the affective or social tactile capacity that could be specific to mammals. Only mammalian species have elongated facial vibrissae, also known as vibraceae, which extend outwards and forwards from the animal's snout. They form a tactile sensory network that envelops the animal's head. Species without vibraceae, or in which they were less apparent, were generally the most evolved representatives of their order. Consequently, having facial vibraceae was an ancestral characteristic of mammals. Many terrestrial mammals, especially those that are nocturnal, live in low-light environments or climb trees, and have evolved highly specialized tactile sensing systems via vibraceae. Research has focused on a variety of these animals, including rodents such as the rat, mouse, and golden hamster, as well as insectivores such as the Etruscan shrew and the Brazilian short-tailed marsupial possum. The base of the Cambrian is marked by the appearance of the Treptichnus pedum fossil record. These are complex fossilized burrows found in seabed sediments all over the world. Treptichnus pedum are thus the stigmata of animal activity on the seabed as they move and search for food. Unfortunately, no fossils of these animals were found in the sediment studied. The mystery remains, all the more so as the organisms responsible for these branching, sinuous burrows are far more complex than the animals of the Ediacara fauna that existed just before. Stages 2, 3, and 4 are marked respectively by the appearance of archaeocyaths, the first reef-building metazoans, and small shelly fossils, which are shell-like microfossils of metazoans represented by sponges, mollusks, annelids, lobopods, and other forms that don't seem to belong to any current phylum. Trilobites also appeared during this period. Then comes the Meolingian period, from 509 to 497 million years ago, which brings together 3 additional stages: the Woolian stage, the Drumian stage, and the Gajangian stage. During this period, trilobite varieties multiplied intensely. Traces of Erectocephalus indicus have been found in Guizhou province in China and in Nevada. Later came Tychignostus atavus, the now extinct trilobite species. Specimens have been found in western Utah. The base of the Gajangian 7th stage is defined by the appearance of the trilobite Lyjopyge levigata, discovered in northwest Hunan, China. Finally, the last Cambrian period stretches from 497 to 485 million years ago, and includes the Pabian, Jiangshanian, and finally the 10th stage, which forms the basis of the period that follows the Cambrian, the Ordovician. New trilobite species continue to appear during this period, but it is the appearance of the conodont, Lapitognathus fluctivagus, that marks the upper limit of the Cambrian. This prehistoric animal has the particularity of having cone-shaped teeth, as indicated by its name, composed of the Latin words teeth and cones. But what might the planet have looked like during the Cambrian period, which marks the beginning of the Lower Paleozoic? In the pre-Cambrian eon, Rodinia was a megacontinent that brought together all the continental masses. Under the influence of plate tectonics, it gradually fragmented. Pieces of continental crust began to drift away across the vastness of the oceans. Shortly before the Cambrian era, two new continents appeared. Laurentia, the larger of the two, forms much of the North America we know today, minus Florida, as well as Greenland and Scotland. The second, less important, corresponds to the Siberian Shield. At the beginning of the Cambrian, a third piece of Rodinia broke off. Named Baltica, it gave rise to present-day Russia, Poland, Scandinavia, and northern Germany. The amputated Rodinia continent was renamed Gondwana. However, it was still vast, encompassing South America, Africa, Australia, Antarctica, Southern Europe, and finally China. As the three newly formed landmasses of Laurentia, Siberia, and Baltica came into being, they were subjected to the effects of plate tectonics. They moved towards and away from each other. Oceans formed while others closed. The Lapitous Ocean opened up between Laurentia and Gondwana when the supercontinent Rodinia broke up. Over millions of years, this Paleo-ocean slowly expanded into the southern hemisphere, thanks to the spreading of the ocean floor in conjunction with a mid-ocean ridge located between the two continents. Around 50 million years after the opening of the Lapitous Ocean, at the end of the Cambrian, a new event gradually modified this part of the globe, reversing the opening process. A subduction zone south of Laurentia, where one oceanic lithospheric plate slipped beneath another, created an island volcanic arc. Volcanic discharges from the island arc mingled for a long time with the great thickness of sand and mud that had already accumulated between the Laurentia continent and this space riddled with volcanic islands. Sediments then accumulated in this zone, leading some 20 million years later in the Silurian to the closure of this ocean, all the more so as small continental masses, also uncoupled from the Great Gondwana continent, slid northwards. Together, these circumstances led to the gradual closure of Lapitous. As the insular volcanic arc moved closer to Laurentia, it eventually collided with the continental margin and began the long process of building a mountain range, the premise of the Caledonian orogeny, which would much later give rise to the Appalachians in North America and the ranges of Scotland and Scandinavia. The Cambrian began as the last ice age of the Proterozoic. The gas-gear's glaciation was coming to an end. A gradual and continuous warming of the climate led to average temperatures well above those we know today. But were the continents thus formed suitable for the development of any form of life? Although the climate seemed favorable, with average global temperatures estimated at 25 degrees Celsius or 77 degrees Fahrenheit, the continents that then occupied mainly equatorial zones were nevertheless, unlike the oceans, devoid of any signs of life. However, Thanks to cyanobacteria, the first algae to appear and play a crucial role on Earth, the great oxygenation of the planet occurred, opening up the field of possibilities. Laurentia, Siberia, and Baltica thus enjoyed a tropical climate. Gondwana, located near the polar zone, experienced relatively cold temperatures, but without a land cover of ice. The intertropical zone, on the other hand, was more arid. The atmosphere contained 20% oxygen and 0.5% carbon dioxide, but the ozone layer was far too thin to protect the surface continents. Relatively exposed to ultraviolet radiation, they were unable to support any form of life. With no plant cover whatsoever, the continent surfaces were totally barren, desert-like and arid. Only the movement of tectonic plates and intense erosion eventually altered the appearance of these lands, which consisted of rocky peaks or barren, cracked plains. The shallow oceans, on the other hand, were able to take full advantage of these conditions to develop the life already present, thanks to their warm waters. At the beginning of the period, only unicellular, then multicellular organisms populated the submarine space. Prokaryotes, single-celled microorganisms with no nucleus or nuclear membrane, such as bacteria and archaea, were found. Then, in addition to the many cyanobacteria, came the eukaryotes, which had appeared during the Proterozoic eon. But we'll see later how climatic conditions played a major role in the evolution of these species over the 56 million years that make up the Cambrian period. Some 600 million years ago, towards the end of the Proterozoic eon, just before the Cambrian era, the Ediacaran fauna, composed of soft-bodied, leaf and tube-shaped organisms, developed for almost 56 million years before suddenly disappearing. The reasons for this sudden disappearance remain an unsolved mystery to this day. However, traces of this fauna have been found all over the world. It was made up of primitive representatives of younger groups, essentially the Cnidarian phylum, soft and medusoid corals, annelids, segmented worms, and arthropods. One of the few survivors of this short period is the jellyfish. But the event that followed was of such importance that it marked the beginning of a new era, the Cambrian. The Cambrian is the era that marks the beginning of fossiliferous times. Following the last ice age of the Proterozoic, the planet underwent a transmutation. Temperatures were much milder, and an unprecedented extension of the epicontinental seas allowed underwater life to develop at an unprecedented rate, but not only in terms of quantity. On a geological timescale, new anatomies appeared on such a scale that we speak of a Cambrian explosion. This was the first evolutionary event of its kind, marking a decisive turning point in the evolution of life on Earth. Some authors have even dubbed it the zoological Big Bang. Most of today's major Metazoan phyla appeared between 541 and 505 million years ago. Although some of these multicellular animals have since disappeared, this Cambrian explosion was the initiator of a great diversification of animal, plant, and bacterial classes and species. living organisms acquired shells, carapaces, and even skeletons, leaving their mark on fossils. Most of these were soft-bodied animals, such as worms and sponges, but the first arthropods, distant ancestors of crustaceans and insects, also appeared. There are also traces of the cephalopods of the future, including the octopus. The rapid emergence of new animal varieties, particularly bilaterians, made ecosystems much more complex than they had been before, when they were populated by relatively simple organisms, as witnessed by the Ediacaran population. Completely new ecological interactions, such as predation, appeared in new marine environments and habitats. Some so-called benthic species lived on the seabed, or even in the sediment covering it. Others, on the other hand, evolved in the aquatic environment. These are known as nectonic animals. The causes of the Cambrian explosion are still not really known. However, several hypotheses have been put forward, some of which are linked to one another. The first is based on an increase in dissolved oxygen in the oceans following the end of the last ice age. There may also have been an increase in the production of phosphate and nitrate ions, essentially for primary photosynthetic production, and enabling phytoplankton development and mineralization of hard parts, such as shells and sclerites. Changes in the food chain, inducing competition between prey and predators, may have favored co-evolutionary mechanisms between different species. As a result, new and vital defense strategies, such as carapaces, shells, and spines, would have developed, offering primordial protection against predators, themselves finely equipped with visual elements. And then there's the question of whether small genetic changes may have occurred accidentally during the embryonic development of certain species. You're probably wondering, and rightly so, about the evidence that has been found from this period. It is indeed legitimate to wonder how fossilized traces of these microorganisms, which moreover lived exclusively on the seabed, can be discovered. If it's hard enough to bring to light the fossilized remains of dinosaurs that appeared almost 300 million years later, what stigmata can be exploited? Fortunately, a large part of the Lapitous Ocean seabed has been preserved. As plate tectonics continually shaped the Earth's crust, surfaces in the ocean depths were raised to mountain slopes. This explosion of life left many fossils that can be found all over the globe. Traces of the Cambrian period can be found all over the world, on every continent. However, current knowledge of life during the Cambrian is based on the study of the Lager state, the storage site still available, where paleontologists are continuing their research. The Cambrian is also well represented in France. Numerous fossils have been discovered in the southwestern Massif Central, more precisely in the Montagne Noire, but also in the Pyrenees, Normandy, the Cevennes, in small outcrops in the Pays de la Loire, and finally in the Ardennes. The first Cambrian fossils to be discovered in France date back to 1888. Trilobite debris was identified in the Montagne Noire, where the stratigraphy subsequently proved to be particularly representative of the Cambrian era. The Burgess Shale is one of the world's most remarkable storage sites. Discovered on August 30th, 1909, by paleontologist Charles Doolittle Walcott, they are located near Mount Burgess, high in the Canadian Rockies, in the heart of Yoho National Park. It's a black shale deposit where fossils, most of them around 505 million years old, bear witness to the end of the Cambrian explosion. Shale is a type of sedimentary rock composed of clay and silt. Often very friable, these rocks reveal the fossilized remains of surprising, even disconcerting creatures. As we noted earlier, the mountains of the Canadian Rockies were once the seabed of the Lapitous Ocean. Sediments were deposited in the deep waters in the form of mud flows, capturing the animals present in this area. The scarcity of oxygen slowed their decomposition, so over time, they were transformed into black, thread-like fossils. Skeletal elements from the Cambrian period are generally small in size. They come from small-shelled fauna. Different parts of mineralized skeletons are studied, such as sclerites, the calcareous part included in the tissues of certain corals, spicules, the calcareous or siliceous constituents, forming the skeleton of sponges, tubes, or shells. These elements probably belong to several different types of animal, but what is most remarkable about the Burgess fauna is the appearance of specific internal and external organs. At last, the animals were equipped with legs, visual attributes, a mouth, digestive system, antennae, and many other special features. To date, almost 150 species of animals, algae, and bacteria have been recorded in Walcott's quarry. Thousands of specimens are divided into several collections, enabling scientists to study the ecosystem as a whole. Quantitative statistics are now possible for the first time, thanks to the sheer quantity of fossils. Researchers estimate that almost 98% of the fossils on this site are those of soft-bodied organisms. The site's unique preservation conditions make it exceptional. Indeed, in most Cambrian marine deposits worldwide, this type of fossil is absent, not because they weren't there, but because they were not preserved in the right conditions. The Burgess community is essentially made up of animals. A few species of green algae and red algae have been recorded, as well as microbial colonies. But the groups most representative of this new environment are undoubtedly the arthropods, those small animals with articulated legs that molt, and the porifers, i.e. the sponges. Without further ado, let's take a look at the species that populated this paleo-ocean, then called Lapitous. Let's start with the most widespread animals, the benthics, i.e. those living in or near the seabed. Canidaspis perfecta is one of the ancestors of the arthropods found today in the form of crustaceans, insects, and arachnids. This specimen, which also left traces in China, measured from 8 to 52 millimeters in length. Its body was partially covered by a carapace and ended in a segmented tail with a telson at the tip, which itself had a double spiny projection pointing backwards. Its bivalve carapace was surmounted by two eyes, between which a spinal column began. It was equipped with one pair of short antennae and another, longer pair, whose function was probably olfactory. Two pairs of legs were located at the level of its head, all equipped with a terminal claw. More than a dozen segments made up its body. The thoracic part was equipped with 8 pairs of associated biramous legs, similarly equipped with claws, while its posterior abdomen was devoid of them. Legs or appendages are said to be biramous when they have both gills and locomotor appendages. Canadaspis perfecta walked along the seabed, stirring up mud with its feet to find its food. Once the food was found, it used its pair of mandibles and maxillae to feed. Among the animals that inhabited the seabed, many species with elongated bodies resembling today's worms lived on the seabed or in the sediment that covered it. Others had a particular morphology that enabled them to swim actively. These elongated animals belong to the category of annelids. 5 species have been recorded in the Burgess Shale, including Piranhacheta, which preferred a sedimentary environment, and Canadia spinosa, which swam with ocean currents. Let's take a look at the Canadia spinosa. This annelid worm measured an average of 3 centimeters or 1 inch long, and could reach up to 4.5 centimeters or 2 inches in length. The main characteristic of this animal is the numerous rigid bristles that extend from its dorsal branches all the way down its back. This feature is typical of polychaete worms. On their distinctive segmented structure, each segment carries a pair of conspicuous lateral appendages called parapodia, used primarily for locomotion. On these parapodia, tufts of a variable number of bristles, the chaetae, facilitate their movement in the aquatic environment. The Piranhacheta, on the other hand, preferred contact with sediments. This tiny, thread-like hairworm takes its name from the Greek words peroni, meaning needle, and kate, meaning long hair, in reference to the bristles that cover it. This worm grew to between 1 and 2 centimeters or 1 inch in length, with a body just 1 millimeter wide without the bristles. Its body consisted of around 25 segments, each equipped with a pair of parapodia. This carnivorous, even detritivorous animal had a straight intestine running the length of its body. Its cephalic region was equipped with a pair of tentacles. This worm was endobenthic and epibenthic, which means it could move both on sediments and within them. Its bristles, very useful for its movements, probably helped it to bury itself in the sediment, both to hide and to search for food. But a much larger worm was discovered in the astonishing Burgess fauna. This is the Capinitator Praetermissus, a worm that proved to be a formidable predator during the Cambrian period. Considered a monster, it measured 11 centimeters or 4 inches, and its broad head was lined with 50 hooks. Nicknamed a Sagittarian worm because of its arrow-like shape, it is described as a chatognath. The name chatognate means jaw hair. In fact, it was its numerous movable hooks that enabled it to capture its prey. This worm is considered a branch of marine predators. Even today, this type of worm can be found in all seas. Although they haven't changed much, they are now often only a few millimeters long. Yet nestled among the plankton, it behaves like a fearsome predator, its hooks claiming numerous victims among the larvae of fish and crustaceans. The seabed was also occupied by brachiopods, animals that fed on suspended particles and whose bodies were housed in bivalve shells. Generally, they attached themselves to a surface on the seabed or to another larger organism by means of their peduncle, a cylindrical, flexible organ. Micromitra burgessensis was one of these brachiopods, whose maximum size was 10 millimeters. Its name comes from the Greek and means little turban. It was the most equipped of the brachiopods found at the Burgess site. It had a mineralized shell. This was adorned with pronounced striations of growth and fine raised lines raising out from the hinge across the entire shell, creating small diamond-shaped patterns on the top. Its valves were subcircular, while its hinge was practically straight. This specimen was also adorned with long, thin bristles that extended well beyond the margins of its shell. These were attached to the edge of the mantle, along the dorsal and ventral valves. Some of these specimens have been found fossilized while attached to the spicules of a variety of sponge, the Piranha sponge, confirming its benthic lifestyle. In the waters of this ocean, animals were actively swimming. This was particularly true of the chordates, animals with dorsal ropes and dorsal spinal cords. Pangaea gracilans, one of the fossils found in the Burgess Shale, undoubtedly represents the most primitive form of chordate. At the time, this ancestor of vertebrates only looked like a worm. However, it has been demonstrated that this animal was equipped with myomeres, the muscles of vertebrates. It measured around 5 centimeters or 2 inches in length. Its elongated body, with a head at the front, gave it the appearance of an eel. Its body, flattened at the sides, suggests that it had a ventral fin located towards the rear. Although this animal spent a lot of time on the seabed in search of food, looking for organic matter, such as snails or sea urchins, it could probably swim at will. One of the arthropods had 2 pedunculated eyes. This is the Sydneyea. With a maximum size of 160 millimeters or 6 inches, it had a convex, cephalic shield, short but wide and subrectangular in frontal view. The 2 lateral corners were notched to accommodate an antenna and eye stalk on either side. Its 2 hemispherical, highly reflective eyes pointed above and behind each antenna. Its thorax was composed of 9 segments, the middle ones being wider. The first 4 thoracic segments had appendages with a large base and 8 lighter ones ending in a sharp claw. The next 5 thoracic segments had identical appendages, but these were associated with branches of filaments, forming what could be described as a wing. Finally, its abdomen was made up of 3 much thinner segments and ended in a triangular telson, accompanied by a pair of large articulated paddles, forming a caudal fan. This carnivore swam and walked on the seabed with the help of its 4 anterior pairs of thoracic appendages, while the filamentous branches of its 5 posterior pairs enabled it to propel itself through the water column. These same filaments may even have participated in the animal's breathing, acting as gills Its spiny basal, coccyx, helped it to crush food and transport it to its mouth. Its eyes and antennae helped it to move above the seabed in search of its prey, which it had to grasp and then crush with its front appendages. Another primitive arthropod had not 2 but 5 eyes. This was Opabinia regulus. Because of its unusual morphology, including a strange frontal proboscis, it is one of the iconic fossils of the Burgess fauna. Its body, whose maximum size was 10.1 centimeters or 4 inches, featured gills and lateral lobes repeated in series, as well as a generously proportioned caudal fan. Its complete body, measured without its proboscis, was between 4.3 and 7 centimeters, or 2 inches. On the anterior margin of its rounded head were 5 prominent eyes. They were bulbous in shape, surmounted by short peduncles. Its ringed proboscis was 4 times the size of its head. Extremely flexible, it ended in a pair of opposing pincers, each with 5 or 6 spines. The ventral mouth was directed backwards. Its trunk was composed of 15 segments, each housing a pair of lateral lobes, themselves bearing gill structures in the form of lamellae. The trunk was followed by a caudal fan with 3 pairs of upward-facing fins. This carnivorous animal swam thanks to the waves of undulations produced by its lateral lobes, which acted as propellers. Its caudal fan, like a rudder, helped it to steer through the water column. Its flexible proboscis and pair of distal pincers were invaluable for catching soft foods and bringing them to its ventral mouth. The seabed was certainly colored by echinoderms. Echinoderms are a distinct group of organisms characterized by a mineralized skeleton made up of several elements with pentameric symmetry. We find this structure today in starfish, whose bodies contain 5 identical parts, radiating from a central point. Naturally, carnivorous animals were fed on mollusks in large numbers. Of the 5 species discovered in the Burgess Shale, only one had a mineralized shell, the Scanella. Sponges also populated the ocean floor. They are among the most primitive animals known to the oceans. Their bodies had no real tissues. Many of these species had a skeleton made up of a mesh of fine mineral rods known as spicules. These demosponges were benthic filter feeders. They therefore belong to the phylum Poriferae. Specimens of several species of the Voxia genus have been found in the Burgess Shale, including Voxia gracilenta. This sponge, very common on this site, measured a maximum of 8 centimeters. Simple and unbranched, or with more complex structures, each of its branches was almost cylindrical, forming a highly accentuated cone with a simple central cavity with a concave or flat opening. To feed, these sessile creatures had no other means than to continually filter water through the channels in their walls in order to retain the particles of organic matter then present. Other species, with variations in skeletal elements and branch shape, were also widespread, such as Voxia bullula and Voxia densa. These could reach up to 15 centimeters or 6 inches in height. Anchored to the seabed, they provided perfect shelter for epibenthic fauna, whose habitat was essentially the seabed. But the best-known sponge in the Burgess Shale is undoubtedly the Hazelia palmata. It is assigned to the upper group of demosponges of the order Monaxidina. The Hazelia genus is highly diversified. It includes species of various morphologies. Some were large and cup-shaped, such as Hazelia palmata, Hazelia crataria, and Hazelia luteria. Others were elongated and conical, such as Hazelia conferta, Hazelia grandis, and Hazelia obscura. There were also some particularly branched species, such as Hazelia delicutala, and others in the form of nodules or lobes, such as Hazelia lobata and Hazelia nodulifera. However, despite the considerable diversity of their general forms, these species all had the same microstructure. They were thin-walled and composed of small spicules densely grouped together in a mesh-like structure that opened outwards. Hazelia palmata, which measured up to 15 centimeters or 6 inches, was distinguished by its open, shield-like shape and numerous clusters of prominent radiating spicules. Among the arthropods active on the seabed alongside these sponges was the Naroya, often compared to trilobites. This marine animal, at most 4 centimeters or 2 inches long, had 2 ovoid dorsal shields with a convex axial region. It therefore had a cephalic shield and an elongated shield protecting the rest of its body. 2 long, articulated antennae protruded from the anterior shield. Behind the antennae were 4 pairs of cephalic appendages, with a further 14 pairs on the trunk. Each of these appendages was segmented and bi-branched. One part formed the locomotor branch, divided into 7 segments and thus perfectly adapted to walking. The other part was a filamentous branch, consisting of a fine shaft from which numerous lamellae sprang. These appendages ended in a large spiny plate. Thanks to its antennae, which were probably sensory, the Naroya was able to locate its food, which it brought to its mouth using the spiny plate at the base of its appendages. It could, however, take short swims, using its filamentous gills to propel it through the aquatic environment. If this carnivore was a scavenger, even a predator, the scars found on the carapace of certain specimens prove that it was also attacked by larger predators. Finally, a carnivore with a tubular carapace completes the list of arthropods. This is the Odaria alata. Most of this animal's body was enveloped by an imposing carapace, the midline of which was visible along the dorsal midline. These 2 united valves on the ventral part make this specimen very special, its carapace forming an open tube at the front and rear of the animal. Its head, which protruded from the tube, had a small anterior plate containing large spherical eyes surmounted by short peduncles. Its body consisted of around 47 segments, each with a pair of appendages. Those of the first 2 segments took the form of slender segmented branches adapted to walking. The other appendages were bi-branched and divided into an inner branch with a large branched basal spine and an outer branch with filamentous lamellae. Its tail, or telson, consisted of 3 fins, extending laterally and vertically. This predator's ventral appendages, blocked by its tubular carapace, probably made it difficult for it to walk on the seabed. Nevertheless, the Odaria was a mobile animal. It could plausibly swim in the water column, thanks to the undulation of its internal branches and biramid appendages. Like a suction pipe, it filtered aquatic organisms that entered at the front and exited at the rear of its carapace. Its large telson, very useful when on the move, helped it to steer, and why not, to manage rolling or braking when swimming. As you can see, this paleo-ocean was teeming with life, even if sea monsters were only small in size at the time. On a zoological scale, the Cambrian explosion led to an unprecedented diversification of animal species. Above all, it marked the emergence of modern ecosystems. The biodiversity of marine life during the Cambrian is also perfectly illustrated in a region of southwest China. Various sites in Chengjiang, located in Yunnan province, are renowned for the quality of preservation of the fossils discovered, including their soft parts. The Chengjiang fauna came after the small shell fauna that marked the beginning of the Cambrian period. New life forms appeared at this time, some 520 million years ago. One of the sites, Mao Tianshan Hill, has been a UNESCO World Heritage Site since 2012. It was on this hill that this lager state was discovered by Dr. Qian Guang Hao of the Nanjing Institute of Geology and Paleontology in 1984. The Chengjiang fossils comprise 16 phyla, over 170 genera, and more than 200 different species. They mark the first appearance of the majority of animal phyla existing during the Phanerozoic, many of which still populate our planet. The first specimens of algae, sponges, brachiopods, arthropods, chordates, and echinoderms have been found. But the long list doesn't end there. Hallucigenia is one of the most studied specimens. This animal, then considered a polychaete worm of the Canadia genus, has been extremely well preserved, providing experts with precious anatomical details. It had an oval-shaped head, attached to its cylindrical body by a constriction giving it a neck. Measuring 22 millimeters or 1 inch in length, it had 7 pairs of dorsal spines and 7 pairs of legs, with small claws at the ends. Today, it is classified as a lobopod. One of the most popular inhabitants of the Chengjiang fauna is a numerous arthropod, some specimens of which are in an excellent state of preservation. This is the Leoncholia illicabrosa. It can be recognized by its elongated segmented body, its biramid appendages, and above all, its 2 large head appendages, each bearing 3 flagellas about 2 centimeters or 1 inch long. Another common animal has revealed numerous anatomical details. This is Urodlichia intermedia, a trilobite that revealed fine uniramate antennae on the front of its head, large crescent-shaped eyes framing a rounded area extended by 2 long spines. Its posterior part ended in a long thorn-like tail. But the most famous fossil in the Chengjiang fauna is undoubtedly the Mylocunmingia vengioa. It represents the oldest known vertebrate. At around 2 centimeters or 1 inch in length, it is a small primitive fish. Its head was distinct from its trunk. It had 5 to 6 gills. Its trunk was equipped with a dorsal and ventral fin, probably twinned. The thorax had 25 V-shaped segments, with the tip turned backwards. While the location of its mouth is not yet clear, it did have a pharynx, as well as a digestive tract that extended to the tip of its tail. Another species stands out in the Cryogenian fauna, this time for its size. This is the Anomalurus sauron. This predator of the now extinct Anomalurus family, also known as the strange shrimp, could measure over 1 meter in length, which is quite exceptional and even spectacular for this period of animal history. Its anterior part was equipped with a pair of large segmented prehensile appendages, edged with spines. Its head was surmounted by 2 large pedunculated eyes, while its underside featured a characteristic ringed mouth. Its segmented body, composed of 11 pairs of lobes, probably made it a very good swimmer. Thus equipped, it must have had no difficulty in feeding on trilobites, which it hunted with a sharp eye. In this complex ecosystem, scientists deduced that over 55% of species were arthropods, 10% were sponges. In addition, more than 10% are still of uncertain form. Most of the fauna lived on the ocean floor. Indeed, few of the specimens we have just observed were pelagic, as their structure was not equipped with elements that would enable them to swim, even passively. Feeding habits were equally varied. There were filter feeders and fearsome predators. While some of these animals were content to scavenge from the sediment, others were active hunters. At the Cryogenian site, the Cambrian explosion is anything but a legend. The diversification of life is well represented, notably with the appearance of numerous phyla, previously unheard of. While many descendants emerged within a single phylum, some endured while others died out in later geological times. Whatever the case, this period witnessed the emergence of all the complex forms of animal life as we know it today. In the lower Cambrian era, during the first 2 series of the Terranuvian and Series 2, a short period stood out, giving rise to the Tomotian fauna, named after the Siberian town of Tomot. This fauna appeared during the Cambrian explosion, 530 million years ago, but lasted only a few million years. Yet, like the Ediacara fauna, it is similarly recognized around the globe. The Tomotian fauna is also often referred to as the small-shelled fauna, and for good reason. While it contains some organisms that look modern for their time, most of its representatives are formed of tiny blades, tubes, needles, caps, or domes. For paleontologists, the nature of this small-shelled fauna remains a mystery. Are they simply scattered fragments of the very first types of shells, still imperfectly formed by unrecognizable organisms? Or are they remnants of pieces of shell that covered previously known but unidentified animals whose construction improved over time? Or do these strange finds correspond to unique organisms that appeared early before spontaneously disappearing? Until recently, the Tomotian fauna was considered to be the first fauna with hard parts. But recent discoveries of organisms with mineralized skeletons have shown the opposite to be true, the latter being found in the stratified layers of the late Precambrian and early Cambrian. Nevertheless, the Tomotian fauna is also important for another reason. It marks the appearance of a group of organisms that most probably belong to the Porifera phylum, that of the sponges, the Archaeocyaths. Although this phylum disappeared at the end of the Cambrian, Archaeocyaths were the very first bioconstructors in association with calcimicrobes, calcifying microbial communities. They were able to generate organic masses measuring 1 to 2 meters in height and a few meters in diameter. These calcified sponges would then adorn the seabed like never before. These massive organisms were the precursors of the complex reef ecosystem that developed a few million years later, toward the end of the Ordovician period. All that remains of the Tomotian fauna is the sponge category. If organisms were able to develop on such a scale during the Cambrian, they were not the only ones. We have already observed a superpredator known as Anomalocaris. This strange, extraordinarily large shrimp, sometimes more than a meter long, wasn't the only one to rain terror on the ocean floor. A recent discovery, published in June 2022, mentions the existence of another giant arthropod, belonging to the Radiodont group. This is the Titanocoris gainaceae. Its remains have just been found in the Burgess Shale fossil formation in British Columbia's Kootenay National Park. Its name, a reference to the word Titan, is self-explanatory and testifies to the exceptional size of this animal. The suffix "coris" means helmet in Greek. At around half a meter in length, the size of this animal was absolutely breathtaking at that time in animal history. While today it may seem almost insignificant, it's worth remembering that at the same period, the vast majority of living creatures were no more than a fifth of this size, with the exception of the Anomalocaris sauron we met earlier. Like its herded cousins, a subgroup of radiodonts, Titanocoris gainaceae was characterized by an exceptionally long head, covered by a 3-part carapace of multiple shapes. The head of this specimen was so disproportionately elongated in relation to the rest of the body that some refer to it as a swimming head. Its mouth was shaped like a cone, with several radiating plates lined with teeth. Stemmed eyes with several facets crowned the mouth. A pair of long frontal appendages, ending in claws and pointing backwards, flanked its head. Finally, its trunk, made up of multiple flaps that were certainly intended to facilitate swimming, supported gills and ended in a front spine. This colossus, whose outer carapace was wider and thinner than that of the average radiodon, must have swum close to the seabed. Adapted to life close to the ground, it was most likely one of the most dangerous predators the oceans could count on during the Cambrian era. Smaller radiodonts, on the other hand, although with similar physical characteristics, were filter feeders. This probably explains the possible cohabitation of these two radiodont varieties, with Titanocoris gainaceae certainly preferring to devour larger prey than its cousins. The study of exceptional faunas such as those of Chengjiang, Burgess, and many others, together with abundant paleontological data, allow us to assert that a complex marine ecosystem newly emerged at the beginning of the Cambrian. While the animal world preceding the Cambrian explosion was already highly diversified, comprising several phyla each, with different strategies for exploiting the environment, the anatomical organization of specimens was still relatively simple. During the Cambrian, we witnessed not only an unprecedented proliferation of living organisms, but above all, the emergence of new anatomical and functional formations far more complex than ever before. From a sensory point of view, evolution is marked by the appearance of eye organs and antennae. Organisms are equipped with gills and a circulatory system, demonstrating that they had a respiratory system. In terms of feeding, complex digestive systems emerged. Some animals possess systems for capturing food particles, and in the case of predators, even prehensile organs. Finally, in terms of locomotion, more or less numerous appendages and flotation organs also appeared. Not to be forgotten are the prominent appendages found in arthropods, some of which have given rise to groups such as the crustaceans. These new anatomical peculiarities are accompanied by an increasingly complex marine ecosystem. Animals are no longer simple unicellular organisms like prokaryotes or eukaryotes, or even multicellular like certain myxobacteria. They are now endowed with a variety of specific organs, depending on which phylum they belong to. The discovery of organisms of different designs indicates the existence of an already highly structured food chain. Indeed, we find preapulin worms inside the sediment, numerous arthropods that lived on the seabed, and even swimming predators such as the fearsome Anomalocaris. But the anatomy of these organisms, miraculously preserved in the fossils, isn't the only clue to learning more about these newly appeared animals. In some deposits, scientists have been able to analyze the stomach contents and coprolites of certain specimens. Observations of the fossilized food in the stomach, and even of the excrement still present in the intestines, has provided relatively precise indications of the diet of some of these animals. In this way, it has been possible to establish that certain species fed on other animal varieties. These stomach contents provide direct evidence of animal interactions within the food chain. In the Burgess Shale, several organic and skeletal elements have been found preserved inside the stomach or intestine of some of the animals studied. The priapulin worm, Otoya, seem to feed on hyoliths, those enigmatic horn-shelled organisms. Several aligned shells were identified in its digestive tract. The arthropod, Sydneia, on the other hand, had a varied diet. Numerous fragments of hyoliths, trilobites, and bradorids, small bivalve arthropods, were found in the digestive tract of this carnivorous predator, which measured between 5 and 16 centimeters, or 5 inches in length. But scientists weren't content to simply observe the digestive tracts of certain species. Among the fossils examined, some could be coprolites. These potential fossil excrements come in different shapes. Some, elliptical and composed of aggregates of skeletal elements, belong to the Chengjiang fauna. They turn out to contain mostly fragments of waptids and bradorids, the small bivalve arthropods that populated the seabed. These are probably excrements produced by one of the largest predators, the Anomalocaris, which measured almost a meter in length. Other clusters discovered in the form of a ribbon and consisting almost entirely of hyoliths are undoubtedly the remains of carnivorous priapulan worms. These observations suggest that endobenthic predators, such as priapulans, were not the only ones to exploit the water-sediment interface. Arthropods, which moved along the bottom, and Anomalocarids, which moved through the water column, shared the same hunting grounds. In addition, traces of attacks and perforations were found on the carapace of some trilobites. Surely inflicted by Anomalocaridids, they testify to the bloodthirsty atmosphere that reigned on the seabed. Fractures and traces of regeneration, certainly due to predation, have also been noted on the shells of an Australian Lower Cambrian mollusk, Marocella. These small organisms, with biomineralized exoskeletons, were similarly subjected to constant predation. The Cambrian was thus marked by the appearance of the first ecosystems with a complex trophic structure, particularly in the animal sphere. Biological interactions, such as predator-prey relationships, are evident. New ecological niches, both benthic and pelagic, were occupied by a variety of predators, including scavengers, detritus feeders, and suspension feeders. Although the pelagic domain was probably not entirely occupied by swimming species during the Lower Cambrian, the concentration of animals in hyperbenthic environments close to the bottom is obvious. This was certainly the first step towards colonization of the water column, followed by the emergence of pelagic food chains. But what were the factors that triggered such an evolution? The establishment of this entirely new ecosystem is based on a number of decisive events and innovations. In the early stages of Metazoan evolution, climate, water, chemistry, and oxygen were most likely important environmental factors. But the acquisition by some species of a more complex nervous system, novel visual organs, and improved motor skills will certainly have enabled the introduction of new selective pressures such as predation. The light switch theory suggests that the introduction of vision into early Cambrian aquatic communities was the main trigger for this ecological renewal. This would have generated a completely new mode of animal interaction and triggered active predation. A cascade of feedback effects followed, such as defensive responses from certain prey, with the appearance of exoskeletons, biomineralization, and even new behaviors. Crucial improvements in the functioning of the nervous system in early Metazoans may have led to the emergence of new vital functions such as vision, limb coordination, locomotion, and even active predation. These new abilities undeniably modified the behavior of fauna, considerably changing ecological rules. Other hypotheses have suggested that major changes in phytoplankton composition took place during the transition from Precambrian to Cambrian, enabling an explosion in animal radiation. An increase in atmospheric oxygen could also have made possible the physical evolution of large animals with higher metabolisms. However, animal biomineralization during this same period is the most important event recorded. Its repercussions on the functioning of early ecosystems can only have been extremely significant. While the Cambrian period saw an unprecedented boom in the development of life, a first extinction followed the Cambrian explosion, which seems to have peaked between 520 and 540 million years ago. But a significant event occurred around 485 million years ago. During this first period of the Paleozoic era, multicellular beings underwent one of the many biological crises that would affect all life forms in subsequent periods. Over 85% of the biosphere's species disappeared. So what happened on the planet to have such an impact on life? While it is acknowledged that 99% of the species that have ever existed on our planet eventually became extinct, the vast majority did so as part of a process of normal species extinction, linked to the limited duration of their biological existence. However, the Cambrian period saw the sudden disappearance of certain species, as did many others that punctuated the development of life on Earth. Some researchers suggest that at the beginning of the Middle Cambrian, the climate underwent rapid oscillations between hot and cold. These climatic changes would have been so rapid that many species would not have had time to adapt, and consequently to survive. A great global cooling and variations in sea level would have modified the terrestrial environment, considerably impacting living conditions on the planet. But what is their theory based on? Geoscientists have successfully dated lavas from the Kalkarindi volcanic province in Australia, as well as over 2 million square kilometers or 770,000 square miles of magmatic rock located in the Northern Territory of Western Australia. Their findings attest to the fact that these lava emissions, which occurred very rapidly, took place at the same time, around 510 million years ago. These events correspond well with the mass extinction of the early Middle Cambrian. Moreover, chemical analysis of these basalts shows that they are relatively low in sulfur dioxide. However, following a major volcanic eruption, an increase in atmospheric sulfur dioxide undeniably causes a temporary cooling of the climate. Could there have been a new period of glaciation, which, although less severe than the Veringer glaciation that ended the Proterozoic eon, would have decimated much of the nascent flora and fauna? Another hypothesis is that the oceans and atmosphere were depleted of oxygen and enriched with carbon dioxide. This global greenhouse effect would have caused global warming. But how did this greenhouse effect come about? Before the Cambrian explosion, the seabed was covered with organic matter that had accumulated over time, forming a thick, dense layer. According to some scientists, the rapid appearance of Cambrian fauna disturbed the tranquil equilibrium of the seabed. The animals, then in great numbers, would have stirred up all these organic layers. By fragmenting and decomposing the organic matter in the soil, the worms and arthropods triggered a mechanism that consumed oxygen and released carbon dioxide. As a result, this explosion of life became somewhat self-sabotaging, depleting oxygen not only from the oceans, but also from the atmosphere. As a result, the anoxic waters decimated the Cambrian fauna on a massive scale, leading to the extinction of numerous species of trilobites. However, geological records show that some species managed to survive, their bodies having adapted to this low oxygen environment. Finally, an extraterrestrial cause, such as a meteorite impact or the supernova explosion of a nearby star, producing a lethally powerful gamma-ray burst, could be the ultimate explanation. In just 10 seconds, the atmospheric ozone layer would have been destroyed, no longer protecting oceanic plankton from the devastating ultraviolet rays that were at the origin of the food chains then in place. A new ice age would then have slowly set in, covering the supercontinent Gondwana at the South Pole with an immense ice cap. With sea levels lowered and ocean circulation slowed, low oxygen levels would have led to much more difficult living conditions. The Cambrian era thus came to an end with a massive biological extinction, known as the Cambrian Ordovician extinction. Brachiopods, conodonts, and trilobites were the animals that experienced the greatest reductions in species numbers. But as with every event of this kind, the surviving species take over the ecological niches left vacant. Nature shows unexpected resilience, and despite the unattractive environment, life continues to thrive. In a much calmer ocean, life returns to normal. Survivors multiply, and new species appear. At this precise moment in its evolution, planet Earth seems to be in full gestation. Will life, which first appeared in an aquatic environment, soon spread to dry land? If we can confirm that this will indeed be the case, will this spectacular evolution of species develop in the next period? For the moment, the Cambrian is giving way to the Ordovician period, a new era in which the global ocean and Earth's atmosphere cooled considerably.